about how to reconstruct metabolic model and different techniques, how you can actually build a very accurate model, and then how you can check your model which is good, which is not good and then it's a iterative process. The model building is a iterative process. Right now whatever information you have that you use for the model, and then slowly you build the model. As time passes you get better version of the model. But how you can use this metabolic model for your experiment? In this sense we will be discussing how genome scale metabolic model can be used for different purposes. So, one of the problem people address using the metabolic model is the metabolic flux analysis. So, the metabolic flux analysis is a technique where you can measure the fluxes experimentally. So, experimentally one can measure the true metabolic state of a system that required accurate flux prediction. This is also a metabolic flux analysis, also a flux prediction, and it actually infer the true metabolic state of a cell or a system you are having it. And then the conventional modeling method are limited in terms of resolution. The flux balance analysis, whatever you learned so far, does not require any experimental data or any experiments you need. You do not need any experiments. That is why your flux resolutions are not very accurate. So, if you want to make your fluxes very accurate, then and some of the fluxes you cannot measure, such as recycle fluxes, bidirection fluxes and parallel fluxes, and the fluxes which has multiple optimal flux distribution. So, those fluxes you need to address in a different way, and that is why metabolic flux analysis is very crucial. The MFA currently measure the intercellular fluxes, and there are a lot of techniques available where you can measure the fluxes very accurately, and it actually calculate the flux in both direction, in the forward and the reverse direction experimentally. And this is the main motivation regarding the metabolic flux analysis when you do the metabolic engineering, or if you want to characterize different kind of cell like cancer cell, HIV cell and so on. Even in the plant system also metabolic fluxes analysis have been done in algae, microalgae, cyanobacteria, in cancer cell line and so on. Most commonly used technique in MFA is basically the 13C level isotope based flux analysis, where you have the substrate as 13C level, and this 13C level isotope, when the substrate is used as 13C level, what will happen? Your metabolite is also level. The metabolites are level, and then you measure those level metabolite using different metabolomics experiment like GC mass, LC mass, and before that people used to measure using NMR. So, these are the three techniques available where you can measure the metabolites, and these metabolites are having isotope level. You know how many isotopes present in the carbon, right? How many isotopes are present there in the carbon? 3 isotopes. The most naturally occurring isotope is C12, and then we have C13 and C14, but their percentage of occurrence is very less, 0.01%. And the good about carbon is that the isotope levels are not harmful to human being. So, if it is harmful, we would not have done the experiment. So, it is naturally present in the atmosphere, we are taking up every day, but we are not affecting. So, that is why this isotope level glucose can be used for the experiment. We do not have to keep any extra protection. And the direct interpretation of the data, 13C metabolic flux analysis not only predict the fluxes, but it actually determine different metabolic pathway. Suppose you are working with some microalgae which we do not know what pathways it involve, completely blank and no genomic info, even sometime annotation is also not done. Then also you can use 13C MFA to find out what kind of reaction it has, and that is why it has a edge. So, this technique is very powerful where you do not know even the central metabolic pathway. So, there you can apply and then you can combine the data with metabolic model for getting the intercellular fluxes. So, in
like if you are using inside the cell, so this will go in every metabolite.
Yeah.
From that, how it's possible to actually identify the unknown pathways, because
I will come to it. How this is all about the method. So, this the 13C MFA has the experimental side and as well as the theoretical computational side. So, it has two parts. The first part is basically doing the experiment, collecting the data and then measuring the data using GC mass or LC mass, and then comes the computational framework. So, the computational framework is very rigorous and most of the techniques came from different optimization method. So, we will learn little bit about the computational aspect of the method and as well as the experimental aspect of the method. The whole MFA came because you cannot measure fluxes directly. So, we have equipment like for DNA sequence you can use different equipment like you can contact Illumina or other company right now available, where if you give the sample they will give the sequence of the entire genome. Similarly, for transcriptomics we have microRNA data where you can actually measure the mRNA level experimentally, whereas in proteomics we have a equipment which can measure the protein level experimentally. Similarly, for metabolomics also we have LC mass where you can measure the metabolite level quantitatively. But when it comes to flux, which is basically the rate of molecule going through a reaction, then there is no equipment which you can get directly from the from your measurement. So, that is why you cannot measure the flux, but you can infer the flux based on the data, based on the modeling simulation. You combine all information, metabolic level and the metabolite data, that will give you the most accurate prediction of the fluxes, because fluxes are the most relevant phenotype. Ultimately, what is happening inside the cell, that determines the fluxes. Even if you measure mRNA level, protein level, DNA level, but it will just give you an idea what is happening inside the cell. But if you can measure the fluxes, that is actually happening inside the cell. When you consider producing some chemical or bioproduct, then fluxes are most important thing. That is why the MFA techniques are becoming popular nowadays. So, the flux balance modeling you have already done. In the last tutorial class also you learned how you can calculate the fluxes, and this is the most popular method because it does not require any experiment, does not require any money. So, people can easily do it, and the technique is that you get a genome sequence and then you annotate and build a metabolic model like this, like you have used K-base yesterday. So, using the K-base you can generate the metabolic model automatically, and you can calculate fluxes using FBA technique. But when it comes to 13C MFA experiment, what you do? You first allow the cell to grow in a glucose. Suppose the cell is growing in glucose. I take the simplest example, but there can be a complicated source of substrate where the glucose is no longer a normal glucose. So, what is difference here? So, the carbons in the glucose are labeled, and you can actually label at particular location. So, the kind of glucose you will be using, that is the isotope level glucose, where the carbons are labeled at particular position. Here, what is the position where carbon is labeled? Can you guess? C1, right. So, you have to culture the cell in such a way that the carbons are labeled fractionally. If it is fully labeled, it will become normal glucose, right. If all the carbons are labeled, then it is of no use. So, you have to label the carbon in such a way that is fractionally labeled, like 1 carbon level. Also, you can use like 20% level or 80% level, like that. You can also first the carbons are labeled and then the amount by what fraction of glucose is labeled, that also you can consider, so that you have a way of tracking the carbon. So, the entire 13C MFA is also known as a tracer experiment, where you track the carbon which are labeled, but you do not track which are non-labeled. Got it? So, this is the technique which is used in many fields, even in environmental science, soil biology, the 13C level experiments are very popular, where you can actually track the carbon where it is going. Basically, the carbons are uptaken by the cell, and then slowly you track those carbon where it is going, and based on that you can identify new pathways, new function of the cell, and so on. And this is the basic idea which actually motivated people to do this kind of experiment. So, what will happen if the cell is growing in a isotope level glucose? What we will see, the slowly the carbon metabolites are also isotope level, and what will happen at the end, you have a almost all the metabolites are level, but we are interested in N metabolites. Can you guess which are the N metabolite inside the cell? Good. Amino acids are the N metabolite. So, once the amino acids are made, the cell uses those amino acids to make protein, peptide, and so on. And in 13C MFA experiment, you collect this N metabolite. So, these are the N metabolite of different 20 amino acid. You can first you collect the sample from the mid exponential phase. So, from the mid exponential phase, you calculate, you take the culture from your flask, and then this cell you can lyse it and get the protein, right, and then protein can be broken into amino acid. So, there are two ways you can actually get the N metabolite, either through proteogenic amino acid or through intercellular amino acid. So, when 13C MFA started long back in 20 years ago, that time people used to use proteogenic amino acid, and proteogenic amino acid can be easily you can get from the proteins inside the cell. If you break the protein, you can use different kind of enzyme to cut the protein, and that will generate amino acid, and that amino acid you can measure in a GC mass. And once you measure the amino acid in a GC mass, for example alanine, you will see that the amino acid is also fractionally level. So, what is this? This is very important, just understand. So, here we have the number of carbons, like 0 means none of the carbons are level, 1 means 1 carbon level, 2 means 2 carbon level, and 3 means 3 carbon level. Since alanine has 3 carbons, we got only 3 peaks, and this is the fractionally level, like 0 carbon level there are no almost 0, whereas first carbon level we have very less, whereas 2 carbon level we have maximum, and then followed by minimum 3 carbon level. So, this is the isotope level pattern you get from directly from the GC mass if you measure it experimentally. So, if it is not level, what would have happened? If I am measuring the alanine in a GC mass, if the alanine is not level, then we would have got many peaks or a single peak? Single peak. Now, since the molecule is level and at a different position, that is why you get a distribution of same molecule, same metabolite is distributed into different leveling position, and these are known as isotopomers. What are isotopomer of a given molecule? It depends on the atoms which are level. We will come to it in more detail, and this is how you can actually measure this metabolite experimentally, and that is used for calculating the fluxes. Up to here everybody is clear?
So, here we are allocating only C13 and C13 carboisotopes and not C12. So, that so that the example.
See, in a GC mass, what is the function of a GC mass? Everybody understand GC mass? It actually measures the mass of a molecule. So, if the masses are different, then it will get splitted. The peaks will be, so each peak is correspond to unique mass of that molecule. So, since the masses are different, because alanine when 2 carbons are level, then it will have a different mass, right. If 1 carbon level, it will be having another mass, and so on. So, 0 carbon level is basically the normal glucose. You are right, the 0 carbon level is basically the normal alanine, where nothing is level. So, and the first one says that it is 1 carbon level, only 1 carbon, you do not know where it is level, the position is immaterial here. Because in a GC mass it cannot determine the location. Can it determine the location? No, it will tell only the mass. The mass of a molecule you can separate, based on the mass you can measure the concentration, and this is basically the fraction of the molecule is basically the concentration, the amount it is formed, and so on. So, the 0 carbon level means this is the normal alanine, and the first carbon is 1 carbon level, and then 2 carbon and 3 carbon, so on. Like that you can measure, and this is becomes your metabolite profile. Up to here everything is clear to you. So, this is the amino acid labeling pattern, you can get it for all 20 amino acid. So, this is glutamate, aspartic acid, alanine, phenylalanine, aspartate, serine, and so on. You can get all the 20 amino acid metabolite labeling. Since the cell has grown on a level glucose, that is why all the amino acid will also be isotope level, and from here you want to actually measure the metabolic fluxes. So, from using this you want to actually go here, and the entire MFA formulation is utilizing this amino acid labeling pattern, and finally it will give you the fluxes, and the fluxes are mainly in the central metabolic networks are considered. Why? In 30C MFA is not like FBA. In FBA, where you have optimization, linear optimization of the biomass. So, you actually optimize the biomass, but in 30C MFA we do not optimize for the biomass, and that is why this is very popular, because as long it is microbes you can still consider considering it for optimizing a biomass, but when it comes to multicellular organism or plant cell, the biomass optimization has no significance, right, and that is why 30C MFA is very popular, where they do not look for biomass maximization. So, there is no biomass maximization in 30C MFA, but there is an objective function, we will come to it. So, this is the formulation of 13C MFA formulation. As I told in the pictorial representation, you have to feed the cell using isotope level glucose, either isotope level glucose or galactose, whatever substrate the cell is consuming. So, you have to make, and this actually costs you a lot. So, this is the cost. So, the level glucose is very expensive, and when it comes to galactose, and it goes more when you consider level galactose, then it is more expensive compared to glucose. And if you want to use any other level glucose, then you have to see whether it is available or not. Not all the level substrates are available, and any unnatural or uncommon substrate, that will be more expensive, because nobody is using that. And then you have isotope level atom with distinct leveling distribution. The distribution I have already shown, where you can actually get the distribution of the isotope level isotopomer in a given metabolite. And this is the overall diagram of the 13C MFA, where you have the glucose entering the cell, and the glucose can be fully level or non-level or 1 carbon level. These are the 3 types of molecule considered in a 13C MFA experiment, mainly these 2, the first carbon level and all carbon level, and this can be either you can choose first of them 20% of the glucose, and this one 80% of the glucose. If 80% of the glucose are first carbon level, then it is 80% level. So, this is the convention followed. Even if you read the paper, you will see this kind of convention they have used. And then once the glucose enter, then you have alanine. So, alanine, if you consider the 3 carbon system, then how many ways you can label? There are 8 ways, 2 to the power 8. If there are in 2 to the power 3, like we have 3 carbons, then you have 8 different isotopomer. The first one can be none of them level, then the second one first carbon level, then the second carbon level, then the last 2 carbon level, and then the first one level. Like that you have 8 different combination for 3 carbon system, and this can be measured using a GC mass. In the GC mass you can measure this kind of molecule, and this peaks like M0, M1, M2, like that you can actually get the fragment intensity. So, in the GC mass what you get, for every mass you get a intensity peak, and it cannot distinguish the isotopomer. All the isotopomer you cannot get using the GC mass. For 3 carbon you can have only 4 peak, because the position. So, this is the degenerate molecule, right. This is the first carbon level, this is also first carbon, this is also first carbon. So, all of them are first carbon, that is why you have only 1 peak. The different isotopomer which are actually having same mass, you cannot separate in GC mass. So, what you require is require a metabolic network. So, either it can be genome scale metabolic network or a small central metabolic network, whatever organism you are actually working on, you have to consider the metabolic network for that system, and then you infer the flux distribution and you apply the steady state mass balance for each isotopomer. So, it is the same concept what you use in FBA, where you assume a steady state approximation. Here also you use the steady state approximation where you have no metabolite accumulation, and then you choose the objective function which I will describe in detail, like how you can actually compare the data between the, it is basically a fitting procedure where you compare the experimental data with your predicted leveling. So, experimentally in one hand you measure the metabolite leveling using GC mass, and in another way you can derive the metabolite leveling based on the computational method. So, and then you compare. So, basically that this is a fitting procedure, and the fitting procedure you have experimental data and the computational data, and then you compare, and as long you go close to very close to each other, then you get a better prediction, and this is the procedure applied in 13C IFA. So, the isotopomer distribution which I have already described can be written in this format, like we have for 2 carbon system we have I00, I01, I10, I11. So, these are the different fraction of the molecule which are represented by IDB. So, one is the mass distribution vector MDB that you get from the GC mass, the MDB, and the IDB you compute theoretically. So, in one hand you have the experimental data in terms of MDB, that is mass distribution vector, and the another hand you have the computationally derived IDV, that is isotopomer distribution vector. So, these are the different fraction of the molecule present inside your system that you can actually either you computationally identify or experimentally identify. Once you have this 2 component ready, then you can compare and see whether it is matching or not. If there is a good match, then you say that flux estimation is done, right. So, that is why this method is very much close to the experimental setup you have. Suppose you are actually studying a strain, and then you actually measure the isotope level metabolites, and then you computationally also get the IDV and MDV, and then you compare, and you when there is a good fit, then you are convinced, okay, I get a better flux prediction. So, this is a kind of reaction you have, like 2 reactant like B, C forming metabolite A, like B has 2 carbon and C has 1 carbon, and then you have 3 carbon, 2 plus 1, that gives 3, and how many ways this can be arranged is shown over here. So, you choose k is equal to 1, 2, and here k is equal to 1 to 4, and then slowly you add it up. So, first case you consider this one, and the second case you consider this 2, like that you have all possible combination, and this is very easy, straightforward, where you get the isotopomer molecule for every reaction. So, that is why it is computationally expensive, like for every reaction it gets splitted into different isotopomer, that means that many metabolites extra added, right. For every reaction, each metabolite based on the amount it is level, we have generating different isotopomer, and each isotopomers are actually treated separately. And this is the mass distribution vector which you get it from the GC mass. So, on one hand you have the IDV and the other you have MDV. So, this is clear to everybody. So, this distribution of the vector or the metabolite leveling you can measure either experimentally or theoretically, and both are required for your flux prediction, and I will describe how you can use these 2 information to calculate your flux. and in order to map the IDV with MDV, we have a matrix. So, the matrix formulation are also, this is also known as isotopomer mapping matrix. So, the mapping matrix, suppose I know the IDV for this reactant, that is S1. Using the IDV of S1, you can calculate the IDV for product. The metabolite labeling we have to calculate theoretically. Suppose you start with your substrate glucose, that you know the labeling, because the substrate you are using for your experiment, you know already the labeling profile, right, and using that labeling profile, slowly you want to calculate for each and every metabolite inside the cell. And for that, if I know S1, I can easily calculate P using the isotopomer mapping matrix, and this mapping matrix is very straightforward. If you make a diagonal and column, you will see that this kind of matrix can be easily formed. It is very straightforward. And the idea is that I want to calculate IDV for all the metabolite inside the cell, and if I know the IDV for the substrate, slowly you can calculate for all the metabolites. And this way, once you know the MDV for the reactant, then you can calculate the MDV for all the metabolite inside the cell, because in the MDV you have to compare with the experimental data, right, and this is only possible because we know the atom transition matrix. Yeah, what is the atom transition matrix? Like for example, we have pyruvate forming acetyl coenzyme A and carbon dioxide, and another reaction we are forming acetaldehyde to acetyl coenzyme A, and these two reaction can be written in a atom transition matrix, that is ABC, that is the how many carbon atoms are there in acetyl coenzyme A? Huh? Huh? Acetyl coenzyme A. So, acetyl coenzyme A, if you exclude the coenzyme A part, because coenzyme A is fixed, right, only the carbons which are present in acetyl group, that is changing. The acetyl coenzyme A, if you consider, then you have three carbon present in three carbon present in pyruvate and 2 carbon in acetyl coenzyme A. So, pyruvate is forming acetyl coenzyme A and producing carbon dioxide. So, this is the simple reaction. I want to calculate the MDV for this reaction.
Pyruvate has 3 carbon, one is in CO2.
CO2, yeah, right. So, you are right. So, this is the reaction, right. Pyruvate is converted into acetyl coenzyme A plus carbon dioxide. So, this reaction I want to calculate the MDV for acetyl coenzyme A. And I know this reaction. So, in 13C MFA, what you do, you choose a flux distribution that can be arbitrary. You just take just a random flux distribution, that is V1, V2, V3, like in FBA you have seen the fluxes, right. So, anywhere you can start the calculation, and this flux distribution, I suppose I choose the flux distribution for PDH, that is pyruvate to acetyl coenzyme A, and ACLDI from acetaldehyde to acetyl coenzyme A to be 0.15 and 0.3. So, this I choose randomly, and then I calculate the MDV for acetyl coenzyme A. If I know the MDV for pyruvate and acetaldehyde, how I calculate the MDV for acetyl coenzyme A? This is very simple, and the atom transition I know. The atom transition is such that the pyruvate, which has 3 carbon, is converted into 2 carbon plus 1 carbon. The 1 carbon is going as carbon dioxide, and this atom transition it has to be very accurate, like where the carbons are moved. Here in this case we see that the first carbon is going out as a carbon dioxide, and today in different databases we have this information where the atom transition map is given, and this is nothing but the atom transition map, where you have pyruvate which has 3 carbon, and then it is producing acetyl coenzyme A 2 carbon and the carbon dioxide, and this way if I know the atom transition map, and for the second reaction it is very simple, the 2 carbon is converted into 2 carbon. So, there is no problem, and this is how you can calculate the MDB for acetyl coenzyme A, provided you know MDB for pyruvate and acetaldehyde. So, this is the MDB you get from the pyruvate, which is already known, because you start from somewhere which is given, and then you calculate the MDB for acetyl coenzyme A, and you normalize using the fluxes like 0.3, which is coming from pyruvate to acetyl coenzyme A in PDH reaction. So, PDH is coming, the contribution, you take this flux and then you normalize for these two, and also for the other reaction the amount of flux flowing is 0.15, then you normalize by the total flux this way. So, this is the technique where you get the mass distribution vector for each and every metabolite inside the cell given the flux distribution. So, this is the flux distribution I assume, right. So, this is the fluxes we assume, and then you calculate the MDB, and this is the algorithm, which is a recursive algorithm where you choose a flux distribution and then you predict the amino acid leveling. So, what did you do in the previous slide? You calculate the MDB for acetyl coenzyme A, and in similar way you can calculate the MDB or the amino acid leveling for each and every amino acid, and this you can do it for a given flux distribution. And in the next step, what you do? You compare with the experimental data. What is your experimental data here? That is what you are getting from GC-mass. What you are getting from GC-mass? Amino acid labeling, right? And that you actually compare. So, in one hand you calculate the amino acid labeling from your theoretical method, because you know the atom transition map, you can get back the labeling using theoretical method, but that labeling may not be accurate. Why it is not accurate? So, the amino acid labeling you are getting here, why it is not comparing well with the experimental labeling? Because the fluxes you consider is not accurate, right. You have just defined random distribution, you do not know the exact fluxes. So, this is a recursive procedure where you choose a flux distribution and then predict the amino acid labeling, and then you compare with the experimental data, and when the fitting is good, that is where you stop. So, there are a lot of equations which actually involved in predicting the amino acid labeling, because this need to be computationally evaluated recursively. So, you can formulate those equations and then calculate the amino acid labeling given a flux profile. For given a flux profile, you calculate the amino acid labeling and then compare with the data, and when the fit is good, this is the error function. In many problems you have seen a error function, right, where you have the experimental data and your computational data, and if the fit is very good, this error function is very small, and that is where you stop the calculation, otherwise it will go back and forth. So, whenever the fitting is not good, then the error function will be very large. That is the difference between the experimental labeling and the computationally evaluated labeling based on the flux profile are very different. If the difference is very high, then you have to repeat the calculation again and again. That is why 13C MFA is very computationally expensive.
So, so is, I mean, it may possible for a given set of experimental data, there are two different kind of flux analysis which are fitting good. Is it possible, or I mean, or
yeah, if your experiment is good and everything is accurate without any error, then you will get a good fit. If there is error in your measurement and everything, you will not get a fit. So, here actually there is in flux balance analysis, you do not know whether the fluxes is accurate or not, whether you are inferring the fluxes is good or not, is very dicey, but here it is based on the experimental data. If the fitting is very good, then you are sure that it is a much more accurate prediction, and most of the time from my experience I can tell you that if the experiment has a erroneous data, then you will not be able to fit that data. So, you will be the 13C MFA experiments takes much lesser time than fitting the data. So, fitting data takes more time. So, you have spent more time to actually computationally fit those data. If the fitting is very good, then you are happy, like okay, this can be very much close to your experiment, and again it is a inference only. So, based on your data, if the data is not accurate or it has lot of, because GC mass will also have lot of erroneous data. So, you have to be very accurate in measuring the metabolite data, and ultimately you get a flux profile like this, and which is a central metabolic network. And 30C MFA is only mainly done for a central metabolic network, it is not done in a genome scale metabolic network, because what you solve those equations are basically a nonlinear equation, whereas FBA involve linear equation. That is why in a fraction of second you get the solution, but in 30C MFA it takes several hours, even with the first stage computer also it takes several hours to solve this problem. So, this is the experimental side of it, like you have the culture like around 5 ml, and then you pallet this culture, and then you extract the metabolite from this cell, and there is a procedure of measure of collecting the metabolite out from this cell, because the cell has many things, right, even the protein, DNA, everything you have to separate. So, metabolite should come out from remaining part of the cell, so that you can do the experiment. And once you collect the metabolite, you lyophilize it and then put it in a LC mass or a GC mass to measurement, for measurement. Once you measure the metabolite leveling and you identify the compound, because in a chromatogram we will have lot of peaks, we do not know where to locate, which metabolite to locate, that you have to identify where to locate, and then you identify the compound, and then the metabolite leveling you can see this kind of leveling pattern you will get, and also you need the concentration of the extracellular metabolite. For 30C MFA you need the fluxes which are going outside the cell, and that you can calculate using HPLC, right. At two different time point you can calculate the fluxes which are going outside the cell, and using these two information you can develop the flux map. So, this is overall the experimental side of it, where you do the experiment, collect the metabolite, measure the metabolite, extract the metabolite from the GC mass data, and then use that information to build the model. And this is the fitting procedure done, like one in the red color you can see this is the experimental data, and the blue color is basically the computational data. So, the experimental data need to be compared with the computational data, and this is a very good fit where you can see is exactly matching with the experimental data, and that is where you stop and you say that this is much more feasible flux profile based on the data you have. This kind of fitting you need to do in order to get a very accurate fluxes. So, what is the difference between 30C MFA and flux balance analysis? So, 30C MFA actually give you a accurate determination of fluxes, because you are using the experimental information where you have the metabolite data to actually constrain your metabolic network. And the most important in 30C MFA is there is no growth maximization assumption. What is the growth maximization assumption? Biomass, you have to optimize for the biomass. And one more important thing in FBA is that you need a biomass equation. If you do not know the biomass equation, you cannot maximize the biomass, right. Here the most important thing in 30C MFA, there is no biomass equation. Is there any biomass equation? No, because you are not optimizing or maximizing for biomass. That is why there is no growth assumption, and this is the most important point in 30C MFA, that is that there is no maximization of the biomass, because when you consider a plant system you cannot do that. When you consider a animal cell you cannot consider maximization of biomass. So, this problem is overcome using 30C MFA, where you do not consider the maximization of the biomass. But FBA has another good benefit thing, it is a metabolite, you can do it for large scale metabolic model, you can go for a very big network, whereas in 30C MFA you constrain your network to a smaller network. So, that is why you can use different predictive method which are already available. you can use genome-wide cofactor balancing, because when you consider a smaller network you cannot do the cofactor balancing, right. All the reaction you have to consider for cofactor balancing. So, there are a lot of benefit in flux balance analysis, and there are also other benefits in 13C. They both are actually very good. So, in my previous slide I work on these two methods, how I can combine these two methods, the flux balance analysis and 13C MFA. So, these are the 13C MFA. You can combine genome scale stoichiometry, you can use the measure fluxes and the 13C leveling info, and this if you can combine both the techniques, then what you got is a 2 scale 13C MFA. So, where you can actually use a bigger metabolic network, and this technique is known as 2 scale 13C MFA, where you can actually go for a genome scale metabolic network, and it uses the principle of 13C MFA, where you consider the central metabolic network for tracking the carbon, and the remaining part of the network are we do not track the carbon, got it. So, it has a different resolution of measurement or calculation. So, when you consider a very big network, what you do? You consider the central metabolic network to be tracked for 13C carbon, and the remaining part of the network, which is a very big network, you can consider as a flux balance analysis. So, here you apply the FBA and here you apply the 13C MFA.
So, how big is this central metabolic network where you can apply?
13C in central metabolic network may be 100 to 200 reaction depending on your network. So, and since you are taking the entire metabolic network, genome scale metabolic network, you can apply cofactor balancing and other techniques can be applied. So, that is why this method is altogether become a different method for calculating the flux 13C MFA for genome scale metabolic network. One more example where 13C MFA, steady state 13C MFA can be used in Synocystis 680, where they have used 13C MFA for measuring the fluxes, where they use the Bg11 media and which is supplemented by glucose and sodium bicarbonate, and they found a pseudo steady state region where they want to measure the fluxes. So, you have to select the time point for measuring the fluxes. So, you cannot do it for all time point. So, we need a pseudo steady state, which is mostly in the exponential phase, right, where the carbons are entering and then biomass is growing. So, this kind of pseudo steady state is required for measuring the fluxes, and this can be obtained in cyanobacteria like Synocystis 6803. In 6803, the fluxes have been determined using 13C steady state MFA. Now this microbial cell which are not going into steady state, there is a different method, that is a non-stationary 13C MFA, which I have not discussed in this class, but some people still think that you cannot do 13C MFA in cyanobacteria using 13C steady state MFA, but you can do it using glucose and sodium bicarbonate, and this is possible. And they got the flux distribution for this cyanobacterial species, where they considered the flux on this reaction to be 100, that is the maximum. You can choose any of the flux distribution to be 1 or 100, and based on that you calculate the fluxes for remaining part of the network, and this is the biomass they have got. So, you can include as many number of reaction, provided it should not be very big, so that your calculation may be computationally expensive. So, depending on the computational power you have, you constrain your network, but if you do it for the central metabolic network, that actually identify how the fluxes are distributed, because the central metabolic network determines how much flux will go in different pathway, and that you can estimate directly from this measurement. Even the method is available for 13C MFA in a microbial community, but here the technique is little different, because in a microbial community you will see that the metabolites inside the culture may not be coming from a same cell. In a pure culture, what happened? The metabolite inside the culture which are secreted are coming from the same cell, because there is only one kind of cell. But in a microbial community, we have a different cell. There is a series of microbes present inside your culture, and in that case the metabolites may not be good to for measurement. So, in microbial community what you do, you measure the peptide or the protein, any small peptide you choose which is very unique. Suppose I say I have 100 organism in your community and you choose a unique peptide based on the sequence that is present only on that organism. It can be 20 nucleotide or 30 nucleotide, depending on your specification, you identify those peptide and then you measure using what is the equipment you use for measuring the peptide or protein? This is popularly known as proteomics. In proteomics you can actually measure the protein concentration, but you can also measure the protein leveling also, and that way you can actually make a peptide leveling profile. Instead of amino acid leveling profile, what do? You measure, you get the peptide leveling profile, and that leveling profile can be used to actually measure the fluxes. So, this is the peptide leveling profile one can get for this sequence like AA, G, AC. So, alanine, alanine, glycine, serine, and so this kind of sequence you can get the leveling profile. So, in the normal amino acid 13C MFA you have a profile for the amino acid, the metabolite profile you get it for the amino acid, and whereas for the microbial community you get it for the peptide, and this peptide can be why peptide is also made up of amino acid which you can break it. So, that information, like you have the peptide leveling, once you have the peptide you can deconvolute the peptide leveling into amino acid leveling, and it is also very easy. So, you can actually deconvolute the peptide leveling into amino acid leveling, and that amino acid leveling can be used to measure the fluxes. So, what are the limitation in 30C MFA? So, you for steady state 30C MFA you need to get a isotopically steady state, that is the isotopically steady state means this cell has to go in a exponential phase where it is in a pseudo, you can get at least a pseudo steady state, and if it is if the cell is not going in a pseudo steady state, then you cannot apply steady state 30C MFA. For that we have a separate method known as non-stationary 13C MFA, INCA, instantaneously non-stationary 13C MFA, and that is also available where you can measure the fluxes, especially isotopically for achieving the isotopic steady state for cases like diurnal alternation in metabolic flux where you cannot get that. So, this kind of cycle you cannot apply. So, in the for that case you have to go for non-stationary 13C MFA, and not very useful for studying autotrophic metabolism where CO2 is only one carbon. So, we need your carbon source to be big like glucose 6 carbon. If it is a 1 carbon metabolite, then also we have a problem. So, that is why when you do flux analysis in cyanobacteria you use both the substrate carbon dioxide and as well as glucose. So, those if you consider only carbon dioxide, then still people get the fluxes, but that has less accuracy. The measurement of fluxes may not be that very accurate, because you have only one carbon system. You got it right? One carbon means all are level. If you, because carbon dioxide is only one carbon, and if you use those carbon dioxide is your only substrate, then you may not get very accurate fluxes. The techniques have been developed to deal with this limit. There are many techniques which are developed to deal this kind of limitation, and still people overcome those limitations to get very accurate fluxes. But the mathematical formulation does not changes. The mathematical formulation like calculating the IDV computationally, then MDV, and then comparing the data, those remain the same. Only in terms of the experiment it changes. Suppose you use a different substrate, then accordingly you modified your method. So, that actually give you a very accurate flux prediction. In summary, the conventional metabolic modeling does not show the whole picture. The conventional like the FBA does not give you the FBA is only applied where you have a very good metabolic network, where you know everything. But if you do not know everything, like whether this pathway is there or not, then 30C MFA is sometimes useful, because because the fitting procedure, if the pathway is not current, you will not be able to fit. That is the criteria used, right. So, if you have included a wrong pathway in your model, then fitting the data will give an erroneous result, and that is where you can conclude, okay, this is may not be a correct metabolic map. So, then you modify the metabolic map and again fit the data, and slowly you go for a recursive procedure to get a very accurate model. So, the many experimental biologists actually corrected different unknown organism by this procedure, where you consider a very small network. You may not consider the whole genome scale network or a central metabolic network, you can consider a very subset of the network, and then you fit the data. If the fitting is good, then you say that this whatever pathway you have chosen is correct, otherwise you change the pathway to fit the data, and this is where you can actually build the accurate model. And you get the fluxes at a higher resolution. So, since you are fitting the experimental data, you get the fluxes at a very higher resolution, and the main concept behind the formulation is steady state flux balances, that is you are not adding any kinetic information. So, you do not include any kinetic data, you just use a flux balance analysis method where you fit the data. So, this is the main idea behind the 13C MFA method. If you have any question, you can ask. So, what is the difference between 13C MFA and FBA? Very good. What are the differences? Very large scale network and cofactor balancing. Since you consider all the reaction, you can do the
Thank you all of you for coming here, and this is an exciting school actually. So, I decided not to teach, instead I will tell a story. Okay. So, as this story has definitely lot of characters and lot of facts. So, as we actually read the story, we cannot remember all the stories, all the facts and the characters, and you do not have to. The whole thing, when we study a novel, we actually remember the essence of it. So, whole idea of this story, if you can get some essence of what I am going to talk about, is beyond how synthetic biology can be used beyond bioproduction. This is that where it can go, where non-genetic engineering cannot go, and you can use those concepts with the bioproduction and do it yourselves. So, imagination is the limit definitely. And if you do not like the story, you are allowed to sleep. Okay. So, in a very short sentence, if you want to describe a synthetic biology, it is basically using the engineering principle in the regime of the genetic engineering or the molecular biology. So, when you call about the engineering, we always look something like this, a very nice building in the Singapore or a rover on the Mars. But you may wonder why you will not see engineering like this. Okay. So, if I ask you a very simple question, that at this technological era, when we could make a vehicle and control its movement from on the Mars, from the Earth with almost a mathematical precision, why we cannot make a Pegasus or the Hawk? Just do not, I mean even if you cannot do a thought experiment, for example, if I say how to make a Pegasus, you do not know where to start. Okay. So, there is a simple answer, and that simple answer is, if you look at this, all this engineering, they do have a well established engineering principle, and based on that you fabricate some of the modules, you assemble them and you test it in the field. Okay. Whereas, we do not know what is the design principle of the life. And in the whole talk we will see that how synthetic biology may help in the future to get the design principle of the life, and what we can do, what the conventional genetic engineering cannot do. Okay. So, let us start a story from a little back, and he is Professor Max Delbruck, who got the Nobel Prize in 1969 for physiology or medicine. He was a gifted physicist, okay, like Amit was a physicist. So, he was also so many of the physicists coming to the biology and actually destroying the biology. Sorry, no, that is just a joke. Do not take it the wise. So he is a gifted. At that time, he is a student of Professor Niels Bohr. Okay, and you can remember at that time it was so exciting for the physicists, because one hand there is a theory of relativity and one hand there is quantum mechanics, and everybody is so excited they are thinking that I can found a new rule of the physics. Okay, and then what happened? He is also not an exception, but what Max thought that if I started studying the biology, then I can get some new laws of the physics from the biology. Okay, but he started with a mindset of a physicist or a mindset of a chemist, and that is why later he is one of the founding father what we call today is the molecular biology. Okay, but when he is bringing the physics into the biology or trying to study the biology from the physical point of view, he also bringing basically the mindset of the physicist, and that is the reductionist approach, which is basically, if you remember in your school days, I mean 10th standard Newtonian mechanics, you can remember the reductionist approach is something you always consider either it's an earth or sun or a car or a ball, you always thinking of a point mass, and then you calculate with the equation, and then again you actually map in to find that. So, that is a reductionist approach. And what is the manifestation of the reductionist approach in the biology? If you look the biology before Max Delbruck or those time and later today, we always, let us say, when you think about a disease, you think about which protein has some mutation, which gene has some mutation. So, we connect a protein with a disease, we connect a gene with a disease. So, that exactly, so we are thinking is this single gene is responsible for disease. So, that is the manifestation of the reductionist approach in the biology, and the highest level of the manifestation is definitely this is the Human Genome Project. You will see that this is not just a scientific project, it is also a political project. Okay, this took like $3 billion that time, and that is why Bill Clinton is actually announcing the success of the first draft of the Human Genome Project in Washington DC. Now, so if you go to the little back on that, and the Professor Watson, in this meeting, Professor Watson is somewhere here actually. So, Professor Watson once actually went to the US Senate, and then he told the audience, that is the Senate, that the disease are connected to the genes. So, if we can genome, I mean if we can actually read all the genes, know all the genes, then we can cure all the diseases including the cancer, and that is the highest manifestation of this reductionist approach in my opinion, and that is gives this very big project, and is a completely an industrial scale project that time. What happened from this before 2000 to today, there is a lot of improvement, and is an accelerated improvement in terms of the technology of this genome sequencing. Now, it's not a industrialist corporation anymore, it's a benchtop machine. Possibly your department has one, our division has one. So, and it's just a benchtop machine. You don't need so many scientists. Any of you, if you trained for 2 months and another 2 months, you can actually do the whole genome sequencing, and the costs come somewhere around $1,000, starting from $3 billion at that time. Now, that gives a big advantage for all others, like agriculture. There is a lot of plant has been sequenced, a lot of bacteria, a lot of yeast, even mammoth has been sequenced. Okay. So, however, so this says that we can read all the DNA, all the genome, but still now we do not have the drug for the cancer. Okay. So, the reason is, we will come to the reason, but if I just want to tell at this moment the reason, so when we think that a biological function depends on a single gene or protein, that is not totally true. Is basically a biological function depends on the network of the genes and the proteins. Okay, that is one of the things. So, anyway, so we come to this point that we can read the things. Okay, Now, can you write? Read means if it is a whole book of the genome, we can read letter by letter. Now, can you write letter by letter? So, that comes in 2008, there is the Craig Venter's group, they actually chemically synthesize the whole genome of a mycoplasma. Okay. So, the writing is different than the PCR, you know, all know what the PCR, polymerase generation, which is nothing but photocopying, because you need a template and you can do the photocopy. Okay. Whereas writing, you can write anything, right. So, this, if I call this artificial life 1, I generally call this as artificial life 2. Just after 2 years, the same group published the paper again in the Science, where they showed they synthesize, chemically synthesize the genome of a one kind of mycoplasma and put it into the another species, from where they actually took out that genome. And when they do that, they change that one mycoplasma to another species. Now, if you want to see in terms of what is the significance, this is not my terms though. So, it is like software is creating its own hardware. So, your genome is a software, because all the programming, all the information is there, and when we are giving to the another species of this mycoplasma, which is, and then it is changing the species means what? It is changing all the metabolites, it is changing the composition of the cell wall, it is changing all these things, even the doubling time and everything is changing. So, that is the hardware you are making. Okay. So, software is creating its own hardware. Now, if you want to see this in a machine world, it looks something like that. Have you seen this movie? Okay. Who haven't seen this movie? I just want to tell you, in this movie, it is one of my favorite movie, and this is architect, which is basically nothing but a program who programmed this whole matrix. Okay. But it is a software. Okay. But this is not only the software, they also make some of the hardware, some of the machines who are trying to destroy the human settlement at the Zion. So, software is making its own hardware. So, in biology, which is practiced for the billions of the years through the evolution, in machine world it looks like a science fiction. Okay. So, now where are we? So, have you seen these movies, either Jurassic Park or Jurassic World or something? Now, if I ask you this question, how to make a dinosaur, can you answer that question? It was in the movie.
Yeah, they put the gene in the some reptiles.
If I take your gene and put it in a toad, it is going to be a Pradipta? No. Okay. So, that is not the answer, that is not the full answer. So, first, if anybody actually read this story, okay, of the Jurassic Park, you will see this exactly, they actually sequence, this is 1986, the story is 1986. They actually, the Michael Crichton read that they found this dinosaur DNA from the blood of a mosquito inside an amber, and then they actually sequenced the whole genome. Okay. And then they synthesized that the whole genome and put it in a frog's nucleus. You take out the nucleus and put it inside the frog, and from there dinosaur. So, that 1986, there is no whole genome sequencing, there is no chemical things writing, you cannot write the whole genome. Here now we have that technology. So, we can read the whole genome. If you get a dinosaur DNA, do not tell Amit, please tell me, so we can make the dinosaur together. Okay. And we can actually synthesize the thing, and then we can make the dinosaur. So, we are at this technology. Now, if I tell you that making a dinosaur is synthetic biology, many people can say yes, I would say no, and we will explain why. So, this is just the dinosaur DNA video where they explained how they get that dinosaur. Now, what we can do at this moment we can read and write the whole genome. Question is, do we understand the book, right? So, here many of you are from engineering or BTech background from IT. So, you know this is a blueprint of a engineering drawing, and everything is written in the English. Even if you can't see it, it does not matter, everything is written in English, all the shapes you know. Okay. So, we can read this, we can write this. Do you understand it? No, right? Okay. I mean, some specialists can definitely understand it, but it is just an analogy to say it is same for the biology. We can read, we can write its information, but we do not know about this, the design. Okay. And the reason it is, I think the Rajib and Amit already told many times that it's all depends on the because of the network. Okay. So, it is very difficult, even he said that the central metabolic network, you cannot do this whole thing for this. So, it is very difficult. Now, if you say this is the computers of the whole world which are connected to the internet, that is a map of 2009, and here is a simplified representation of a biomolecular network of a bacteria. If you just see in these two, you can just by seeing that the level of the complexity is same. If you tell a mathematician or a physicist to decipher that, they will generally use the topology. From the topology, what you can tell, for example, this part is the, you can see the higher concentration. If you map the world map, maybe you find this is India. Okay. So, many Indians are using the Facebooks, and if you come here, you won't get much, maybe this, I do not know, maybe let us say Nigeria. Okay. Not many people are using, you can, this kind of things you can tell. This is the same for the protein networks, when we generally not the very specialized people, but general people, when we look at the protein network, we look at this. So, this network may be responsible for the cancer, this, that is a static picture, but that is not a biological function. When it comes to the biological function, and most of the time it's, there are three things are involved. One is all those interactions are basically non-linear, the gene regulation interactions are non-linear. They have a lot of feedbacks with each other, and there is a lot of noise, and when it comes to, there is no mathematics available to decipher that. So, from a whole network of a biological network, when you consider all the dynamics, there is no mathematics, no technology available in this world at this moment which can even tell of a single biological function, okay, not from this way. But that does not mean we are not going to try. Okay. So, from the end of the last century, people have started understanding that there is some human knowledge base. That knowledge base always deals with this network, noise, and the feedbacks, and that is basically the electrical or electronics or computer engineering. So, that there could be one way to understand the biological system from that perspective, okay, and you can loosely define this as a molecular systems biology. So, if this philosophy is correct, then another philosophy is, then it is should be correct that we can also develop a small biological network by following those engineering principles and plug into the cell or organism to change the function predictively or actually develop a new function. Okay. If that philosophy is true, this philosophy could be, and this philosophy is loosely defined as the synthetic biology. So, whole idea of the synthetic biology, let us say develop. So, if you follow all the rules of a computer system, where those physical layers are coming to the gates, and they have a principle to make the gates from the modules, and then computers and the networks. So, what about if we take the proteins and genes, we develop some biochemical reaction which follows the same principle as those gates, and the way these gates are connected to the module, if I can develop the pathways, okay, from those biological reactions following this principle, not what of the inherent biological principle, then we may come to some point that when we develop some of the new functions of the cell or completely new function, even completely new organism. So, this is the whole philosophy at this moment for the synthetic biology. Now, all of you know that this is the one of the first example of the synthetic gene circuit, or which we call the synthetic biology, and this came from the James Collins group, where they have developed a system, simple plasmid, okay, and if you give the IPTG, it is a small molecule chemicals, and then the green fluorescence protein is increasing. You give an anhydrotetracycline ATC, it is coming down. Now, you may ask you the what's I mean what's special about it? Those kind of gene regulation you are always getting in any undergraduate biochemistry book, but the catch is something different. So, think about this laser pointer. When I press my finger, you get a light. If I take my fingers off, there is no light. Okay? We call it switch. In engineering sense, this is not a switch. Now, think about those light switch of this room. Okay? Now, do a thought experiment. You press the switch, you turn the light on, you take your switch, your fingers off, the light is still on. Okay? Similarly, you press it to turn it off, you take your fingers off, still it is off.
So, those switch has a memory, it could remember that you pressed. Okay. So, a simple switch in engineering switch must have a memory, but this is a knob, this is not a memory. So, exactly what they have done. So, they put an IPTG and this green fluorescence protein increases, they took out the IPTG from the system, still this EGFP is there. You take this ATC, it is coming down, you took the ATC out, still it is off. So, what they have developed, that memory, okay, that is the most important. But how to make that? So, if you want to construct that, first what you need a theoretical principle or a qualitative principle, which is based on this RS latch, and in biological regime it looks like two mutually repressing promoter gene pairs. But even if you take two mutually repressive gene promoter pairs, you cannot come to that switch. The reason, it requires a quantitative principle. This quantitative principle only tells only at the certain parameter range this will give the behavior of a switch or the bistable behavior. Any other places it will work like a knob. So, when you have this quantitative range, then you can actually whatever the synthetic gene circuit you are trying to make, you can actually change the translation rate, transcription rate, protein DNA interaction, and then come to that range, so that you can make. So, this is a fundamental difference between the genetic engineering and the synthetic biology. In the synthetic biology, first you need a principle, a qualitative principle, based on that you are trying to put your biological stuff. Then you need a quantitative principle from where you get your ranges, or you get a mathematical model that how this system will look like, and you assemble and you get the desired behavior. So, that is the engineering cycle to make synthetic biology. Now, it is like this computation in the biological cell is definitely not news, that is something we are trying to do. The cell do the computations for a long time, okay. There is memory and everything. One example I can give, have you seen this movie? May or may not be, I do not know. I do not like this movie, but I just want to say a similarity between this guy and this guy, okay, through the movie, not the guy itself. So, if you remember, even if you do not remember, it does not matter. So, at least you know this guy have a transient memory, right. So, what I can remember a sequence when there is a he is chasing a person, chasing, and suddenly there is a big truck was coming and actually hit the guy whom he was chasing, and that point Amir Khan actually forgot everything and he looked just perplexed what he was doing here, right. So, what it suggests that if you want to act, you have to remember your previous thing. So, I am chasing someone, I am just remembering I am chasing, I was chasing, right, and suddenly when it is blank, if this memory is blank, I do not know what I was doing, right. It is the memory and it is coming from the head. Now, think about this bacteria. This bacteria, if you have a high school biology, you do know the bacteria has something called chemotaxis. So, what it does? Let us say do a thought experiment. May I take this board for a moment. Okay. So, you have a bacteria and here let us say is a sugar cube, and you put it is the whole thing is a beaker, just assemble, and this is my way of drawing bacteria. And so, which means the concentration of the sugar is high, very high here, and it is going to decrease that side. So, if bacteria is there, it has some receptor, it sense that sugar and it is going to come in this direction by swimming, okay. It has the flagella. Now, the bacteria is just a 2 micrometer to 5 micrometer, and this is the even if it is static, there is always a Brownian motion of the water, which makes this bacteria to flip this other way around. Now, if this bacteria goes in this direction, just completely 180 degree direction, it stops, it do not start swim. Again something gives it heat, it comes to this direction possibly, it start going there, again something heat, it starts going, if it goes it stops, and so on. Now, that means it is coming in this point, it stops. How it stops? Because it is sensing less sugar in this part. So, it cannot swim on that, which basically means it has a transient memory, because if it does not have a transient memory, it could not differentiate between the concentration of this previous instance to this present instance. Okay. And this is not as simple as the switch I showed, it has a much more complex, and there is a lot of control theory involved here. But what I am trying to say, memory does not mean you have to be head. Okay. This is a molecule can do that kind of the memory. Okay. Many people like me who does not have a brain, but have memory. Okay. So, anyway, so okay, I put okay, thank you. So, if that is a possibility, now I will give you one example, this only example I will try to give in little detail. Okay. So, that you to know that if you want to make a gene circuit, how you can do that. Okay. So, I will tell my experience, and that is so. So, when the synthetic biology is coming, so there is a joke about synthetic biology came. So, for example, it is about an antivirus software for human. So, for example, one day morning I have antivirus software in my body, and one day morning I wake up and suddenly say, hey Sangram, you are possibly contaminated with a Nipah virus from the heritage of this IIT, and if you want to debug your system, you go to this store, you buy those softwares and debug your system. So, that is a joke about the synthetic biology, but we can do for the human cell definitely, but it is not advisable to do that for whole human. There is a lot of ethical issues, but what we try to do is a very simple system, and that is in the bacteria. So, this is the bacteriophage lambda is a virus which attacks the bacteria E. coli and writes its genome into the chromosome of the bacteria and stays dormant generation after generation. This is called lysogeny. Now, if you give UV radiation, this actually there is some biochemistry changes, and this virus starts copying itself like crazy, destroy the bacteria and so on. Okay. So, what we have tried to do, we have tried to develop a synthetic gene circuit and put it in this bacteria. Now, even if you give the UV radiation, it does not go to this lytic cycle, but at the very same time it follows all the properties of antivirus software, like it has to be dormant at the normal condition, it should automatically detect the viral attack, it should automatically turn on prevent lysis, it should mute because it is a device, you can reset after it works, and most importantly it has to be mathematically predictable. Okay. So, this thing is very well studied, this lysis and lysogeny for the gene regulation came, the idea of the gene regulation came from here, lot of physicists, mathematician, chemists actually studied that. So, we know all the molecular components, we choose a molecular component, basically part of the molecular components which can be, you do not have to remember, but which can be actually explained in terms of the nonlinear dynamics. Okay. And what we have used is some theory of the control. I just want to see that let us say you have a system, and in terms of the biochemistry, when it is lysogeny phase, there is some protein which is the high concentration, and you give the UV, there is some disturbance, there is something happens in the biochemistry, that protein starts cleaves. Okay, and it reduces the amount of that. So, what we have developed some of the synthetic controllers. It has two parts, one is the sensor and actuator. The sensor sense that decrease of that protein, okay, and then start producing that, the actuator start producing that protein and actually bring it to the set point. Okay. Now, it has to do fast enough. That is why the synthetic biology, the dynamics is very important. It is not you cannot just use a linear equation to solve all those, because dynamics, why it is important, it has to be fast enough before this what is called this lytic cycle takes over. If its lytic cycle takes over, you cannot do anything. So, this controller has to be produced, go to the set point very fast. Okay. So, with that this is the biology, you do not have to remember all this, but what we have to also make a mathematical model. This mathematical model says only when there is a green part, only when the green, this system is going to work, any other red it is not going to work. So, that is the parameter range, and based on the parameter range we have developed 8 of different controllers. One of them actually works very well for all of this. This is just experimental proof, you do not remember, and this is the match between the mathematical prediction and experimental results. 22 condition after 25 actually matched. So, it is a good match in terms of a biological system. Okay. So, there is a lot of examples, you do not have to remember, but Similar way you can fight different disease. For example, this is a sense and destroy pathogen. So, you engineered your gut E. coli in such a way when there is a increase of a pathogen and they send some molecules, that molecules is sent by your engineered gut pathogen and they secrete one peptide and which actually kills that particular pathogen. Okay. So, it is the sense and destroy. The similar thing can be done on the uric acid where there is a disbalance in the uric acid homeostasis that can be sensed by some cells and that cells produce that particular enzyme to make that. So, it is the sense and then produce, sense and cure. So, this is the kind of a approach of the program therapeutics which you cannot get in the conventional therapeutics. Okay. So, sense and then you do something. Okay. It is autonomously you are doing it. This is another very nice example. There is no practical application of this example though. So, but because they developed a classifier circuit inside a cancer cell and if you put this classifier circuit based on 1, 2, 3, 4, 5, 6 mRNA level, they can actually identify this is a HeLa cell or not. If they identify this is a HeLa cell, they produce a protein and actually goes to the apoptotic pathway. Okay. But you cannot use in reality because it is as a cell level, you cannot put the whole all the cells of the human. You cannot do that, but this is this is a very complex circuit, this is a very very complex circuit in that range. There is other things, this is like a programmed pattern formation. There are the two different kind of the cells, one cell is producing green fluorescence protein and another red, but all those pattern is mathematically predictable and based on that you can make these patterns. They have a genetic half adder, there are pulse counter and so on. There is a lot of things are coming. And now this is an interesting example, bomb detecting plants. Think about a plant which I put it here and then someone is coming with a bomb and this plant from green goes to white and you can say, hey, this guy has a bomb there. Okay. So, now how do you know how the bomb squad, there is a dogs, how they find there is a bomb and we cannot find? Do you know? Smelling? Yeah. So, there is nothing in this world is basically totally insulated. So, whatever the bomb you want to make, always it is some chemicals are coming out, the dog can actually smell that. So, here whole idea was, but this is not this actually happened reality, right. So, here you engineered the a plant which is called Arabidopsis in such a way that it can sense that TNT, yeah, it is not RD, it can sense the TNT, trinitrotoluene, okay, and then it basically there is a repressor which cut out the chlorophyll forming circuit, and then it comes to green, white. So, this is looks like that, in the machine this looks like that, but definitely it is not instantaneous, it takes somewhere around for more than 24 hours, less than 48 hours. So, that way you cannot detect a bomb, right, but you can think about the other applications. For example, the many places in the Africa they have the land mines. So, you put a tree which can actually detect the land mines or the environmental pollutants, you put it here and some environmental pollutants coming here from Kolkata or something, you generally know that. So, those kind of applications or many other kind of applications you can think of. But what I am trying to say, this is possible, okay, that is all. Now, I come to this slide is made by my student Kathagoli. Actually, this is the difference between the genetic. Now, at this moment at least you come to the position what is the difference between the synthetic biology and the genetic engineering. Now, I remember 2 days back someone is telling about the BT cotton. Okay. So, BT cotton is definitely, I mean, if BT cotton is synthetic biology, then what synthetic biology is doing, because this is genetic engineering, people know that genetic engineering for a long time, and whatever today we have is just because of genetic, different kind of foods to medicine and everything is just for the genetic engineering. Okay. So, for example, this is the golden rice, you have heard about the golden rice which has the Indian scientist made, where you put the keratin gene inside the rice and you get the golden rice. So, you can get the vitamin A or keratin in that way. But we are not getting a golden wheat or golden something, right. Why is that so? because making it is not that simple. It is not just you take a keratin gene and put it and everything is starting giving, like Pradipta was saying, just take Pradipta's gene and put it in a frog embryo and it looks like a Pradipta. That is not going to happen. Okay. So, that is not going to happen. So, this likes a lot of expertise, a lot of trial and error, because you do not know what is going to happen. So, that those are the genetic engineering. On the contrast, the synthetic biology, what they try to complement, this is synthetic biology is not the champion. Synthetic biology is just to complement that. If we come to a position where we can actually design everything in the beforehand. If I want to make a fruit which has the like I do not like a fruit which is in Bengali called sobeda, okay, but if anybody put the flavor of the mango, I definitely like it, okay, but you really do not know how to do that, okay. So, if there is a predictable way to do that. So, basically genetic engineering plus engineering design principle gives to this. So, this kind of complex circuits you cannot make through the genetic engineering, you definitely need the help of the engineering in the level of the biology. So, that is at this moment, that is the fundamental difference between the synthetic biology and the genetic engineering. Now, what you can do is just I will give, let us say what we are trying to develop in our lab. I am not going to the results, just what we want to do. So, you have possibly heard about the micro robotics. So, the micro robotics is making the robots is very smaller scale, and so the lot of robots can actually communicate with each other to make some of the tasks. So, these are the robots people, the swarm robots, this people already made. This is from the Sangeeta Bhatia's lab, and this is the artist impression of the nano kinds of robot or micro robots of the future. Now, this micro robots is possible because in electrical or electronics engineering, now people can put a very complex controller in a very small chip, okay. That is the whole idea of this. So, a lot of kind of chip you can put this kind of robot, and you can lot of sensors, lot of things you can put, and that is the thing you can do in the micro robotics. The question is it is also possible to do the soft robotics, and that is what we try to do. So, whole idea is the bacteria, or bacteria is simple to work. So, that is why we work on the bacteria. It is a cell which is 2 to 5 micrometer. So, exactly the size of a micro robot. Now, if we can develop very complex artificial signal processing systems in the bacteria, then we can actually turn this bacteria as a robot to do different kind of work. Now, I will show you that how synthetic biology can do to help you. You can do your robot, okay. We are trying ours, you can do yours. So, what is the challenge? Like when the synthetic biology first started, like in 2000, the switch on different kind of gates and come to the counter. If you want to make a robot, you have to go to the level of this complexity at that range. You cannot just from the switch, you cannot make a robot. So, you have to come to this range or somewhere here, okay. So, at least here. So, we are trying to develop, for example, it is called a priority binary encoder, that is a very important circuit in the computers, do lot of things. Our so if you want to do in a biological regime, it is here all this D1, D2, this is a line, this is basically the electric line, this is electric flow or not. Here you can have 4 different extracellular chemicals, it could be any chemicals, but we are trying to do based on these chemicals, and then based on you have different active line and output A1, A0 in terms of different fluorescence proteins, okay. I will come to how we can actually develop that. Similarly, we are trying to develop decoder. The decoder has 2 different inputs, and for each of the combination it can have 4 different outputs or 4 different decision lines. So, here you can decode some extracellular signal by a biological system like a cell by producing 4 different proteins, and if you want to make exactly you want to make like this. Now, the thing is, so why synthetic biology is different than the biology and engineering. So, if I ask you to make a truth table, a system of a system like this, it will behave like this, and if you start thinking in terms of the promoter genes and the proteins, okay, it is almost impossible to come out of a solution, okay, at least for me, okay, at least for me is impossible. But if what you can do, if we can make some of the gate, like for this an AND gate, an inverter and different kind of NOR gates, OR gates and something, then we have this electrical engineering principle which is going to tell you that if you connect those gate in this way, then you are going to get this kind of behavior. This is going to be an encoder, and where if you connect this cell like this way, you are going to get decoder. Okay. I do not have to think the inherent biology of the system, and that is the essence of the engineering. When like that is why a Mercedes car, okay, can be I mean a mechanic of a 8 pass, they can actually work on that, right, because there is a very inherent design principle. You know, this module will have to come out, this module will have to go, and so on, so on. Thus that is the idea of the synthetic biology, to make the biology exactly like engineering, where you do not need to think about all the inherent thing, but just follow the engineering principles. That is why if you want to make this kind of room, any BTech graduate can actually design this kind of room or any building, okay, or even a chemical engineering graduate can develop a photoreactor, okay. But if I say tell a biology student to make golden rice, impossible. You need a lot of understanding of this biology. So how we take out the understanding of the biology that level from this engineering level, that is the goal of the engineering. Okay. So forget about this. Okay. So when you make a circuit, it is not just you connect this thing, as I said. There are 3 things. If it is a Boolean circuit this kind, you have to think about the 3 things. One is that it has to be its dynamic range, its digitality, and how it is prone to noise or not. So these things, if you control these 3 things, then you can say this circuit is robust. For example, here we try to develop simple AND gate based on 9 different designs, and only these 2 is giving better behavior in terms of the dynamic range or the fortunes. You see there is a lot of leakage here, this leakage has gone, this leakage is still here. Okay. So this is the leakage and that is the inherent, as Rajib already said, that inherent is the leakage in the biology, and that is the problem with the promoter when you actually doing the metabolic engineering. So this is the leakage, but these are better than this. So this is the dynamic range, but just these 2 looks almost same, but that does not mean they are AND gate, because AND gate is a digital. So you have to come, I am not going to the details how you are thinking, but what you can see just looking at these 2 gates, the one gate is better digital. What that means, like there are 2 signal axis IPTG and ATC, here you can see it is suddenly it comes up from a plane, right, whereas here it is coming up gradually. When it is coming gradually, that is not digital. When it is coming a sharp transition, that is the digitality. So you have to think about the something if it is digital or not. And third is the noise. I do not have the experiment on that at this moment, so I cannot show, but you have to also look at that if what is the noise or variability on that. Forget about that. Now what we are, if there is a, so this is all possibilities you can do in different way. So if we can really make those robots, okay, I mean at least some artificial signal I have to do, otherwise my student do not get a PhD. So definitely we make at a certain point, but what we are going to do with that, okay. So one possibility you can think about, this is, I do not know if you have read this is the fantastic voyage by Isaac Asimov, and this movie is very old movie, 1966, and in this science fiction what happened, so in a patient there is a deep wound somewhere, very small and deep wound somewhere where the surgeon's scissor cannot go, okay. So they have a technology to miniaturize all those things. So they miniaturize all the surgeons and nurses, doctors, put it in a submarine, miniaturize, and put it, just inject in the bloodstream. You can see these the red blood cells, okay. And then the doctors come, find out which is the disease part or which is wound part, do the operation and came back. Okay, definitely we are not trying to do that. Okay, you can do that, but what we can do that, what we are planning to do, and it is the work of my 2 students Rajkamal and Dipro, where we want to look at the cancer microenvironment. So when the cancer drug delivery happens, basically cancer drug delivery, nanoparticle based deliveries and all those things, they always try to target, or even chemotherapeutic delivery with some ligand attachment, the receptor on the cell, okay. Now what is uncultivated is that the microenvironment, because microenvironment has a different kind of metabolite signature, it has low oxygen, pH, ROS, inorganic phosphates, lactate and different, which are different than the healthy environment. However, the point is just by a single parameter you cannot differentiate cancer and non-cancer. For example, if all cancer tumors are hypoxia, that does not mean any tissue which is hypoxia is cancer. Okay. But when so there is a signature of the chemicals, but there is no way to target those chemicals. Okay. So, what we are trying to develop, we are trying to develop a lot of sensor and lot of decision making circuit inside. So, that based on this, in a cancer mimetic environment, this bacteria decide if this environment is cancerous or not. If this is cancerous, they produce a protein which will help this bacteria to invade the cancer cell, okay, and actually deliver the drug, which is basically in our case is the shRNA. Okay. So, that is the goal, that how can you do that. So, this is where maybe the ROS robotic platform can work, and hope at least something we have to get those guys get PhD. Now, that is so robotics, many people are trying robotics in different way. So, you can think in one way. And another very important part I would say where again genetic engineering cannot come, and that is a particular part of the tissue engineering. You have heard of the tissue engineering. So, tissue engineering is the making artificial organs outside the body using the cells. The artificial organ like the dialysis, what people do, that is not an artificial organ, that is basically the membrane, that is basically inorganic things you are using, and here is the actual organ. Let us say you want to put a heart transplantation. Okay. At this moment you need the heart from a human body, but the whole idea of the tissue engineering, can you develop that heart outside the body. Okay. So, that you can do that. Now, the way tissue engineering grows, so people can make from, for example, very recently people made the heart muscle tissues from the stem cell, the skin stem cell, they change it to the heart muscle. That is a very important discovery definitely. But what you cannot do from these cells, you cannot make a heart, okay, because heart has a shape, it has something inside to function. Now, the way people try to do tissue engineering, they use some material scaffold, okay, which has the structure of a particular organ like a kidney, and then you try to grow the cells so that it gives some of the kidney of kind of the structure. Okay. So, that is the one way, but it is very difficult to the material scaffold a organ like heart. Maybe you can do urinary bladder and those kind of, but heart at this moment of the material science and engineering is impossible. Okay. So, here synthetic biology can give some lead. The reason in our body when we grow from the cells and how our heart grows, there is no material scaffold. Our cells have that programming through which we make the heart, we make the kidneys and so on, right. How much time do I have? 10 minutes. Okay. So, now the principle of, so if you want to develop those kind of the heart, the principle coming from the physical chemistry, very nonlinear dynamics and physical chemistry, the first, which is this is something called BZ reaction, and this is like 1900 something, and people never looked at it, but this has a very interesting thermodynamic function. So, if you just mix these four things together and it gives you a color like a purple color, okay, and you just in the beaker you stay there and you start the reaction and just stop that stirring, and it is like this. Now, you see after 10 seconds it is coming blue, and then again it is coming as a purple and again coming. So, this reaction is oscillating. This is different than the chemical reaction we always studies. So, it is A plus B and C, when it goes to the C equilibrium it stops, but here the reaction is oscillating. Okay. So, that is people did not understand in terms of the chemistry what is the use of it. So, anyway, so this is stirred, same reaction if you do unstirred. So, if you put in the petri dish, do not start that reaction, you will start getting pattern like this. Okay. Now, if I give this, do you know who is this? Alan Turing. Alan Turing. So, Alan Turing, apart from this father of the computation or Turing machines, he also proposed something, it's called reaction diffusion system, possibly many of you may know. So, this reaction diffusion system with a reaction, if there is a controlled diffusion, means one part can go diffused but other is not, which is always happening in the cell, some metabolites can go, some protein cannot go, then it can form a pattern, and he thought that in the developmental biology like in the fly, those patterns are coming from this reaction diffusion system, and later, lot later, like last 10 years, possibly people can prove using the reaction diffusion theory that this kind of pattern is coming from this reaction diffusion theory. Okay. So, here is just example, the digit formation. Digit formation means when, so this we have this 5 digits, right. So, it is not forming in a one day. Similarly, so you are coming to a pattern. So, that patterns in the rat is coming to the reaction diffusion theory, and this is the gene circuit, inherent gene circuit. This is not a synthetic gene circuit. This gene circuit along with diffusion theory is giving this kind of the pattern. Okay. Now, if you think about your developmental biology, the heart, the whole thing is the pattern formation. The first thing you have to do a 3-dimensional pattern forming. Through that you can make that heart inherently the way it is in our body. So, this is like the pattern formation in the bacterial cell in the 2D. Okay. This is synthetic gene circuits, and later this is 2005, this is I think 2015, after 10 years the other group actually. So, here you can see 1, 2, 3 concentric cycle, and here you can they actually increase that to the more, right. They are coming towards how similar to this pattern formation, but in a 2D format using the inherent programming of the biology. People try to do the 3D pattern forming. Here they are separating 2 cells, one is going towards the outside and another is coming to the inside. That is inherent pattern forming. You just mix these 2 cells and you do the some gene circuit, and they are doing in that way. Okay. Similarly, other kind of pattern formation in the 3D gene circuit. So, this is going in the first step to a different way to look at the tissue engineering, not the materials way, but the totally synthetic biology way where genetic engineering cannot come. So, this is one way the synthetic biology can lead, and you can think of this kind of things in your for future things. So, in last 5 minutes I will just try to say another way where synthetic biology is very important, and that is the space exploration. Okay. Now, space exploration is different than the space exploration what is in the 70s. That time human has to show they can go to the moon. Today when we are trying to go to the human subject are going to try to the Mars, the idea is not to show that I can go to the Mars. The idea you have to make a research base there. Okay. And going there it takes around 2 years, and coming back is around 2 years. So, this 4 years in the space is completely hazardous, and you cannot take all the resources from the earth. So, if you want to have a some biryani, okay, you cannot take it from the Aslan. Okay. So, you have to actually make the biryani, even the rice and everything on the Mars. Okay. So, all the resource what Mars has, you have to use those resources and to make for that. So, what is the solution? How we are going to do that? What NASA thinks synthetic biology is the way. It can take all the resources whatever here has, and you can starting from biomaterials, starting from the road, all the environmental system, and everything can be done using the microbe. So, one is so why synthetic biology could be the key to the space travel. People think do not pack everything you need for the space mission, just pack enough bacteria to build things for you. Okay. So, that is the essence of the space synthetic biology, if any of you are interested. So, it is not just for the Mars, but for something else. So, if you grow the cell, okay, without any programming and put it in the space, they will form a 3-dimensional structure. Okay. So, this is you can say 2-3-dimensional structure, you just put the cells in the space. So, if in terms of the tissue engineering, this is also very important. Okay. There is one slide I did not, so I just cut because it is something else, because space is not just. So, for example, Intel when they make those chips, okay, they can make only a small silicon. Okay. The reason silicon is so fragile, its gravity will actually break it. So, they found, and another thing is that you need a very clean room, okay, because in a circuit the difference between one wire to one wire is 21 nanometer, and there is just a dust comes on, everything is gone. So, if you go to the space which is totally clean and you can make it, and the cost they have done a cost estimation, it is 27% less than doing in the earth. Okay. So, it has different way of also engineering values of the space. So, anyway, so if you want to so here okay, I will just try to finish up. So, microgravity is also hazards for the space, because there is a lot of disease comes from the microgravity like heart disease and stuff, but this can be explained from the fluid flow in the zero gravity. But there is also reduced immunity. People have tried to understand that reduced immunity in terms of the cell molecular genetics way, and there is some process to do that, like differential gene expression and the passive mapping. It do have a two caveat. So, the caveat is when you analyze on transcriptomics data, here say 7% of the Huntington disease genes have been altered. Does that mean Huntington's pathway is altered? You cannot say, because there is no guidelines to say like 70% of the cells has to be 70% of the genes have to be involved, or 7% is good enough. Okay. So, that is one caveat, and another caveat in terms of the network, because in those experiment microgravity is very low perturbation. So, there is a network may be perturbed, but the whole physiology has been perturbed, but not a single gene which is perturbed more than 2 times. So, you are missing in common method. So, so my student actually developed a different systems biology pipeline using some machine learning algorithms and some running some statistics, and analyzed 8,000 molecular pathways based on all the space data what available to NASA, and we got good results. I am not going to the results, but it is Sayek's work, and they got something in the newspaper. It is good for them also as a new lab, because at least something you can connect with the common people, that is science, otherwise it is very difficult to connect with the common people, and if it is a newspaper it is always good. So, after his publication they got like so we got a lot of disease signatures which people do not knew the before, and that is the thing, and it is also for the bacteria. Okay. So, here I am not going to details, but what I am trying to say is a lot of metabolic pathways has been altered which cannot go through the conventional way. But now if you want to think some metabolic pathway in a microgravity reactor on the space, then you have some lead from just a transcriptomic analysis that the metabolic how is changing. So, you can actually take that data in the cellular context to whenever doing some metabolic engineering models, and the ultimate goal of this is definitely make a designer organism, something like that. Okay. And this is for the energy science, because Professor Srivastava told me that whatever you tell it has to connect with the energy, and I am connecting energy in a different way. It is not necessary you have to produce the lipids and the biodiesel and fuel in the cell. There is another way to look at the energy problem. If you have you seen this movie in Avatar? So, if you can make a glowing tree and put it in the IITs, okay, you do not need this night lights. Okay. So, it is, and you do not need to get these lipids and things back. So, those you do not need this flux analysis. So, you can do that. So, this is another way of the thinking what you can do. So, possibilities are enormous. Okay. It is, and you are very young. So, you may think about telling whatever your background does not matter. Okay. So, my PhD supervisor was a space engineer. He designed the first that robotic communications on the first rover's Mars mission. So, and he come to the synthetic biology. So, it is totally exciting. Okay. And okay, forget about. So, just acknowledgement my students, my funding, and definitely my gurus, Christopher Love at postdoctoral supervisor MIT, and David Macmillan at University of Toronto. Thank you very much. If you have any question.
Any question? Very interesting talk about synthetic biology. You have not about protein-protein interaction. He is one of the coordinator of the course. We both of us actually designed this course actually for you. Hope you enjoyed this last talk. Thank you.
Okay, thank you, Amit, for the introduction. Okay. So, I got this is the first opportunity to thank you for participating in this GAN course and making it successful. Okay. So, I have 2 lectures. One is here on protein-protein interaction, and after the lunch there will be one tutorial or hands-on session for sequence alignment. So, in this lecture I will try to unfold the story of protein-protein interaction. So, as you are aware about the fact that most of the functions are performed because of the interaction of one protein with say another protein or DNA or RNA or small molecule. Now, being a computer science student, my background is computer science. So, I always try to look at the tools or the softwares which is used for several purposes. So, here today I will talk on this protein interaction. So, probably you are aware about lot of softwares which are there for protein-protein interaction, or sometimes that is called as the protein docking, and some softwares are there for network analysis. Probably you heard the name Cytoscape software. So, how they are written? So, my intention is that what is going on inside the tool or software, I am interested to know that one, with an intention that if there is any requirement, I will customize that software for my own purpose, and for that I need to know the working principle behind this one. Okay. So, here I will try to introduce you with this. So, just to refresh our memory, this is very basic introduction. Okay. An amino acid is composed of a protein is composed of a number of amino acids, which you can consider as a basic block. Here is the structure of an amino acid, where the central is a chiral carbon atom, and 4 hands of this carbon is hydrogen, amino group, carboxyl group, and the 4th one is a R group, which is a variant. By replacing that one with different AH or CH3 etc., we will get different amino acids. Now, if this amino acid is placed one after another during the synthesis process, then what we can notice that this is a carboxyl group, this is the amino group. So, one hydrogen from here and one OH from here is being out, they are forming one water molecule, and at that time there is a covalent bond between these two, and because of this bonding, one amino acid is connected to the next, to the next, to the next, that way there is a connection and we get long chain of protein. Okay. Now, this amino acid, there are 20 essential amino acids. Mostly they are represented by single characters A, C, D, E, etc., alanine, then cysteine, then aspartic acid, then glutamic acid, etc. Now, corresponding to each amino acid, if I replace them by that single character, then I can write one single character corresponding to this amino acid, another single character corresponding to this, third, fourth. That way I can have a long string or sequence. That sequence is called as the primary structure of that protein, and which is looking like this. Okay. Now, if I replace each amino acid by the constituent atom and allow them to be in 3-dimensional space, then corresponding to each atom I will have one x, y, z coordinate, right. Now, if I replace them by that x, y, z coordinate, then it will take some shape in 3-dimensional shape. Okay. So, from here I can get one tertiary structure for that one. In 3-dimensional space, what I will get, that is the tertiary structure. Now, you can think that okay, this is a glycine. I am not sure whether you can read from here glycine. So, that is the simplest one, where in the side chain there is only hydrogen. So, if you keep on replacing them and then try to allocate some coordinate corresponding to each atom, then the possibilities are enormous. Now, among all those possibilities, which is going to be more stable is determined by several facts. Now, when we do experiment, then we do not much care about that one, we just tell that whether the protein is stable or not. If it is stable, then using some experimental techniques we got that. If it is not, then we are not getting that one. But if I look from computer point of view, or if I try to develop one algorithm where this will be taken as an input and I wish to have this as an output, then that particular problem is called as the, you heard the name, protein folding problem, right. Lot of people are working since 1970s to develop some software. Several softwares are there, several competitions are there to demonstrate that whose software is best, blah blah blah. Okay. Now, given one tertiary structure of a particular protein, it should not have a function. It will have a function only when it will interact with other protein or DNA or RNA or some small molecule, and because of that interaction some function will be there, and when they will interact then we will get one structure that is called as the quaternary structure. Okay. Now, if I ask you that in hemoglobin, okay, how many such proteins are participating in order to form a quaternary structure? 4, right? 2 alpha, 2 beta, like that way. So, if you are given more than one tertiary structure, generating the complex out of that is called as the protein docking problem. So, basically in protein docking you are trying to understand that if these two protein interacts, then what is their interacting point. Okay. Now, you understand.
So, like when it's a quaternary structure coming from tertiary structure, so that is also the part of the protein folding problem, or it's
No.
generally separate?
This is protein docking problem, this is protein folding problem. Okay. Now, as you know that hemoglobin has a specific structure. If the structure is lost, then among all those properties like oxygen transportation etc., its function will be lost. So, given 4 say different chains of the hemoglobin, if you are asked to develop some software or write some algorithm through which if you connect them, then you will get a complete structure of hemoglobin, and if there is any mistake then you will not get the required function. So, that is the catch line. It is not the case that given a input I am giving some output, but if the output does not have any function, then basically it is useless. And in that way also you can think that the possibilities are enormous, right. So, simple example I can say that if say this is one protein, okay. So, this is a say primary structure, this is a tertiary structure I got because of some protein folding problem. Okay. Now, this is another protein, I wish to determine whether they will interact or not. Now, the possibilities are like they can interact here, 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 in all possibilities are there. Now, among those possibilities which are feasible or which are going to be more stable, we need to decide that one through this protein docking problem. Okay. Formally, if I define, then I can say ab initio protein-protein docking is the determination of the molecular structure of complexes formed by 2 or more proteins without the need for experimental measurement. Of course, without the need of experimental measurement, because if we do experiment, then that is the final test. So, why do I go for this protein docking? Okay. Now, few more points will also arise from here, that why shall I go for development of such a software? Because as of now what I am telling, if this is one protein, this is another protein, then this is the quaternary structure. That structure I am able to determine at the atomic level resolution, right. Now, how many different experimental techniques are there through which I can get atomic level resolution of a molecular structure? X-ray crystallography, NMR, cryo, right. Now, all those have some limitations, right. So, for X-ray crystallography it must be soluble, NMR also it is true, right. Now, the amount of time taken for the crystal process and the rate of success etc. involved with that one. So, combining all those things, the time and cost factor is very high for all those experimental technique. Okay. So, this is giving you one alternative. So, what we are telling that computational technique is giving you an alternative corresponding to that experimental technique. That is why it is without the need for experimental measurement. Okay. Now, before going into details of that one, so let us start by looking at this. Given this as a protein and this as a protein, now here you can see this is one protein and this is another protein, right. Now, I need to identify the complex, means initially each atom has one coordinate position. Now, after the complex formation I will assign some new coordinates with that, right. So, for example, this was the coordinate 0 0 0, then 0 0 1 etc., here, and this was something. Now, after that assignment, I can consider that I can keep this as fixed, and then I can take this only, and then I change the coordinate of this one and identify what is the best possible fit. Then I am done, right. While I am doing that one, then if you look closely to the atom, then you can see that you did not have to look for all those atoms which goes inside during the protein folding, right. So, like look, this is the primary sequence, let us assume. Because of the folding, the structure has become this. Now, because of the folding, structure has become this. Now, when I am developing algorithm, then the interaction will be here. Interaction will not be in this region, So, what I can do from the list of atoms in red color and blue color or participating molecules, I can straight away keep all those atoms which goes inside due to the folding aside. I am not telling I will remove those, I am not considering those for the time being. Once I will fix the coordinate of the surface atoms, then I can accordingly adjust, because then it will be just a transformation matrix multiplication in order to get the complete structure. That way also I am doing one thing, I am reducing the size of the problem. So, initially say this protein has say 2000 number of atoms and this has 2500 number of atoms. Now, whatever computation I do, okay, so if I have to do it on 2000 and 2500, and if I consider only the surface, which may be say 700 or 800 and say another 1300 etc., then that is a huge reduction, right. So, computation time will also be less, right. So, I can consider that one. Next problem which will arise is that if you consider that when say I am telling you that corresponding to each atom there is a coordinate, coordinate is just a point mass, right. So, this is a coordinate, this is x, y, z, but actually if we consider, well, for the easy calculation, that it is not an atom is not in wave form, there is no electron proton, but just it is a hard sphere. That approximation we can do based upon the van der Waals radius. Okay. Then I can say that this is one sphere. Okay. Now, what I am with, given input of these two protein structures, okay, I have actually only set up atoms and corresponding to each atom I have only coordinates. Well, I can have the radius also, and using that one if I do, then I will get something like this. From here, as per my previous assumption, I can say that okay, ignore this one for the time being, rest of the surface atoms. Then the question comes, how can I represent the surface of a molecule? Okay, because these are the coordinates. Okay. Now, how do I know that who is inside, who is outside? That is one point. Another point is that although these are the points, but I cannot say that say this is from red and this is from blue. Okay. So, two molecule when they will interact, if they are not like this, then there will be a clash, right. So, as if one atom is trying to penetrate another, and if it is the case, then we know this graph, right. You remember this graph, Lennard-Jones potential, right. So, when I will try to go inside, then I will get a very high positive value. So, it will not allow. So, that is not going to be a correct coordinate assignment. So, first of all I need to know that how I can basically represent the surface of a molecule. So, there are a lot of work on that one. people have worked on solvent contract, reentrant surface, parts critical point, grid representation and internal coordinates. Let us try to look at that. So, these are the atoms. Okay. One method, what it tries to do, that it is taking another small atom, you can consider that as a sphere, and here these atoms are also sphere. Then it tries to roll that sphere or the atom, which I can call as a probe, over this, and if you basically roll over this, then what will happen? You look, in some position this surface will be in contact with this surface, in some position it will not be in contact with this one, right. So, that way what we will get, that some region where there is a contact, some region there is no contact. So, it is defined that this part where there is no contact, it is the reentrant surface, and where there is a contact, that is the contact surface. Now, actually if I wish to measure the molecular surface, it will be the sum of the reentrant surface and the contact surface. Okay. Now, the question is, what is the advantage or benefit of having this proposal? It is very simple. Most of the time protein or any molecule is in some solution, right, and solution means, if I consider it is water, then I am only interested about those atoms which is accessible by the water molecules. Isn't it same as rolling the probe of a water molecule on the surface of the protein molecule, right. If I do not consider that it will get wet, the molecule, then, and if I consider that my solution is with color red, and if I put my molecule inside that one, then at the atomic level, so all the contact surface will be colored with the color of the solution. Oops, sorry. It will be colored by the color of the solution, and in this reentrant surface I have to do some corrections. In this reentrant surface I have to do some corrections. Why? If I do not, then I will miss this region, because up to this it is accessible, up to this it is accessible, this much is not accessible. Now, by combining those I can get total molecular surface. At the same time I can able to understand by the say coloring that which atoms are on the surface. So, all those atoms which are not on the surface I will ignore for the time being. Okay. So, this is one representation scheme, and here is much more elaboration. So, just look at this, this is the atom from the protein, this is. Okay. Now, when a probe or say water molecule rolls over this one, then it is rolling like this. Okay. Now, last time I said when there is a contact surface and this is the reentrant surface. Alternatively, I may think that if I look at the trajectory of the center of this probe, then I will get this blue solid line. Okay. That is called as the solvent accessible surface. Okay. So, from application point of view, you may think that there is, although from value point of view there will be a little difference that you can understand, right. So, which will be higher, solvent accessible surface or the molecular surface? Solvent accessible surface, it will be higher, but the calculation is very fast for this solvent accessible surface compared to the molecular surface. So, whenever you need to go for say high throughput application, say you wish to calculate the surface area of say 10,000 molecular structures, then probably this will be your better choice compared to the previous one. Okay. Now, another representation says that okay, these are the atoms on the surface, I understand. But if I look at the biology, as of now I was not consulting with the biology, but now if I consult with the biology, I may get some interesting point, that even I did not have to consider all the surface point. If I have sufficient experimental or say literature information corresponding to the binding site, I may consider that okay, these are important, rest are not. Then again I can have another subset. Okay. Alternatively, what we can think that when say that information is also not there, then this is forming almost not exactly a straight line. My drawing is not straight line, but this is straight line of course. Okay. Now, whenever this is a straight line, I may think that okay, this molecule has a shape, and when I am considering the shape, then if I consider all the atoms where on the left and right and in 3-dimensional space in all the neighbor cell there is a change of the inclination, then I will consider only those atoms. So, for example, this and this, then I can capture the shape information more correctly. So, here during that time my intention is to reduce the number of atoms further, so that my computation will be fast. Okay. From that point of view, you may think to have a better surface representation where you may not consider all the points, okay, but say I am considering only the yellow colored point or red color point, which can depict the total shape of the protein. But probably the simplest, as I like, is the grid representation. What you will do, that you put the molecule into a grid. In 2-dimensional it will be looking like this, although this grid is of very higher length, but you can decide on this one. Okay. You can consider that this is my origin, right. Now, along the x axis, say in each 1 angstrom you should have a vertical line, and along the y axis in each 1 angstrom you have a horizontal line. Okay. Then what you will do, that you will check from this coordinate whether an atom belongs to one cell or not. If yes, you flag it 1, if no, you flag it 0.
What is the optimal dimension of the grid?
So, there is no optimality, you can check based upon the length size of this protein. So, in all 3 directions x, y and z, you can check minimum and maximum.
Each cell.
Oh, this each cell? Okay, generally 1.8 is considered, but you can change that one. Okay. If you do that one, think it is nothing but you are moving from analog space to digital space. Say for me, if you consider that I am a protein, okay, then I am a protein, my photograph is taken. Now, if you look at that photograph is nothing but a matrix. So, there you will find in some pixels I am present, in some pixel I am not present. If you write one simple step and implement that one, that if I am present it is 1, if I am not present then 0, then you will get a binary matrix indicating that my presence. Now, using this if you do, then you will get one more advantage there, that if one atom is present 1, if no atom is present then 0, and if it is inside, using that surface analysis I can identify that which atoms are inside. If I say that is very high positive value, then you see that if I multiply each pixel, then what will happen? So, 1 multiplied with 1, that is 1, right. 1 multiplied with 0, 0, right, and 0 multiplied with, so 1 multiplied with 1, 1, 0 multiplied with anything, it is 0, and rho multiplied with delta equals to very high value. Okay. So, let me write something delta delta, okay. And if I multiplied rho with 1, that is also rho delta 1 delta. So, what you can see, 1 means if I say that it is on the surface 0 means it is outside and rho means very high value and it is inside, then 1 multiplied with 1 will indicate when the surface of this guy will be overlapping with the surface of this guy, right. That is allowed and that is what I am interested to look at. When 0 will be multiplied with something, then it is 0 means there is no overlapping with each other. I am not interested because it is not forming any complex, right. And when it will be rho or delta or say delta delta, that indicates very high positive value. What is the meaning of that? There is a class, right, because it is going inside. So, that is not allowed. Now, after doing this one, if you say this is corresponding to one cell in my grid, if you keep on summing the whole matrix, then you will find one value, and if I say that is a score value which indicates the amount of overlap if it is not in this region. Okay. And if you consider this one also, this is just nothing but a penalty which will give a very bad measure. Okay. If I say that this is of negative value, okay, then it will go down. Now, if you sort them in the descending order, then on top you will get the maximum overlap region.
So, these grids are 2-dimensional grids or 3-dimensional
No.
grids?
So, this is just for the presentation, it is 3-dimensional. So, there is one more advantage of this one, you can basically implement in fast Fourier technique, which is another computational technique I am not discussing, but if you do that one, then again you will get some speed up. Okay. So, we have represented the molecule in the surface and to some extent we are able to identify that what is the amount of overlap. Okay. And definitely our intention will be to maximize the overlap. Maximizing the overlap indicates the maximizing the interaction surface, right. Now, if I say that my intention is to identify this region, then I am correct, right. During the protein-protein interaction, given red and green color molecule, I am interested to identify this surface only. Okay. So, in order to do that one, first what you need to do, as I said, if this is a protein, this is another protein, you have to explore all the possibilities. Okay. After exploring all those possibilities, then you have to rank them among these which may be feasible. Then among those feasible, you have to score them based upon some scoring function. I will come to that scoring function later. Okay. Now, you can consider as if the docking is this stepwise thing. So, first you got a molecule, you have one surface representation technique using that one, and by generating all the possibilities you got some feasible cases, and from there you will score and rank them. Now, during this generation all the possibilities, you should keep one thing in mind, that if you generate too many cases, then that is advantageous. Why? Because if say this is your solution and you generated 3 cases, one is this, another is this, another is this. So, in your generation there is no solution. So, there is no way that you will get the solution. So, during that generation your intention will be to generate as many possibilities as possible. Now, I can call all of them as decoy. Decoy means it looks like a solution, but it is not actually a solution. Okay. Now, if I look at the crystallization process, then docking is of 2 kinds when basically we do this process. So, why it is? So, if say one complex is there which is co-crystallized. Okay. So, for example, I am taking one hemoglobin structure, and that is a crystal structure where 2 alpha 2 beta both are present. So, all 4 are present. Now, I am then extracting 1 alpha, 2 alpha, 3 alpha, 1 beta, 2 beta, and then I am trying to do the docking. Now, look, during the crystal process they are optimized, right. Now, if this is their co-crystal structure. When I am extracting them out, this and this, then if I allow them to generate all the possibilities, then one such possibility will be created, and look, this is most optimum fitting, right. On the other hand, if I crystallize this separately and if I crystallize this separately, now look, when they are co-crystallized, these are optimum adjustment between these two. Now, if this is separately crystallized and this is separately crystallized, there is no guarantee that it will be look like this. It may be like this, like this, like this, like this. And if say this is like this, I try to fit, the fitting will not be proper, right. From that point of view, first one is called as the bound docking, when you are dealing with the co-crystallized thing, and second one is called as the unbound, when they are separately crystallized. Okay. Now, you have to deal these two things separately, because for this, when they are co-crystallized, so after determining their decoy structure, you may not have to adjust this, but for the second case, when they are separately crystallized, say one has become this, another is this, they will not fit, but they will fit only when you will allow them to relax. If they relax, then they will fit. So, one extra step will come for this unbound docking, which sometimes is also called as a predictive docking. Why it is predictive? Because these two are separately crystallized. So, you do not know what is the complex structure, you have to predict this, but for the first case you know the complex structure. One more thing, mostly people do the rigid body docking, means throughout the simulation or algorithm development, they consider protein as a rigid body. So, as such, the limitation is because of the computation time. If you consider that it is a flexible body, then say for one atom, if it is free, then 6 degrees of freedom are there, right. But when they are part of a molecule, then few degrees of freedom are restricted. Okay. Now, if you consider that it is flexible, then the number of degrees of freedom will increase, and if I consider all the possibilities, then the computation time may increase. There are several docking search techniques. Okay. People have tried different techniques like pseudo random, directed search, brute force search. Okay. Among all those techniques, this brute force search ensures that all the possibilities are generated, but this pseudo random or directed search will give you only a subset of this brute force search. So, you may lose some true positive case or the solution during these two steps. However, the advantage of these two different kind of searching techniques is that they are very fast compared to this one, because the number of decoy they generate is less compared to this one. So, these are just for the reference, I am not going into details. So, people have tried a lot of things. You just look at the year, right, from 86, I do not have the latest, but when I did PhD, then I found that in each month about 3-4 different techniques are coming up. So, people are working either on docking strategy or on scoring functions. Okay, still the accuracy is not that much good. Now, here once we will have a set of decoys, how do we score them in order to get the proper rank solution? So, grossly the parameters which will be used for the scoring is divided into this category. One is the geometric complementarity. It comes from the lock and key mechanism of the biology. So, as we know that when two protein interacts, mostly there is a lock and key mechanism. Okay. But this is just an idea. When we are developing the algorithms, we need to quantify what do you mean by that complementarity, right. So, similar to that, we know that hydrophobic residues are trying to go inside during the folding and also during the docking. This is just an idea. How do you quantify that? Several hydrophobic scales are there. Using those scales also, if you try to do, then you will see that some hydrophobic residue might be on the surface. It is nothing but an optimization problem, where say if the length of your protein is 100 residue, among those maybe 60 or 70 is hydrophilic, then 30 or 35 is hydrophobic. Okay. So, all those hydrophobic may not be at the core, but they will try to align in such a way that maximum of the hydrophobic residues goes inside. But again the problem is how do you quantify that one, but because the moment you will try to develop an algorithm, you should have a deterministic equation. So, some mathematical equation, once you will have that one, you implement and you get some result. Okay. So, for each of them you have to have, but the justification is very clear. For this geometric complementarity, it is the compactness. For this intramolecular or intermolecular overlap, I will not allow this kind of overlap because of this Lennard-Jones potential. Okay. For hydrogen bond, this is very trivial. If the number of hydrogen bond increases, then it will add some extra stability on the complex structure. Contact area, increase in contact area ensures the interaction will increase. Very small contact area mostly gives either transient or no interaction. Okay. Then pairwise amino acid atom atom contact, electrostatic interaction which includes the Coulomb interaction. Okay. Along with that one, solvation energy is also being considered. So, people have a different set of features, but grossly you have to consider all these. So, here it is a very simplistic model where the first is the interface area, which is you can consider as a contact area also. The next area is is the interface packing, which ensures that how much packing is there, because if this is the case, so here there is a void, here there is a void, and you are trying to form one complex and this is the contact area, then it is not going to be much stable, because there is something going to be inside. Okay. So, if it is the case, then there is a problem. So, you have to find out that how much packing is there. Okay. Then you have to find out how much complementarity is there. Okay. So, this is very complementary, okay, but if it is, then the amount of complementarity is less. There is no such complementarity in this case. Okay. Next is non-bonded interaction, which includes two parts. One you can see that is the Lennard-Jones potential, the attractive and repulsive part, and the third one is the Coulomb interaction, where you need to assign some partial charges to each atom, and then you can calculate that how much interaction is there. So, overall that is the non-bonded energy. The last one is very interesting and also the equation. Okay. So, it says that one complex will be stable only when that in the individual cases the stability is higher compared to when they are forming the complex. Okay. So, this may be the case if I consider that this is in water, this is in water, then the amount of interaction it is doing with the water. Now, if they are forming the complex, then the amount of interaction it is doing with the water. So, this complex is more stable compared to these two individual cases. So, this term is mostly captured by either solvation energy or desolvation energy term, but I like this equation. It was published in 1986, long back in Nature. So, one guy David Eisenberg, so what he has done, so using a handful of proteins, some 8 to 10 protein structures, he did one experiment and gave one very simple equation. You can see that this is the linear combination of a number of things. Okay. So, this is the summation of atom type, this is delta sigma is basically one feature, and this delta ASA is the amount of accessible surface area or surface accessible area which is lost due to complex formation. It is very easy to calculate. Now, during this atom type, what he has done, if you remember that in the amino acid carbon is one part. So, carbon goes to one atom type. Another, nitrogen and oxygen being part of charged residue or being in the main chain, right. So, nitrogen is there in amino and oxygen is there in carboxyl. Again, nitrogen is there in lysine and arginine, and oxygen is there in glutamic acid and aspartic acid side chain. So, these two oxygen and nitrogen are not same, right. So, that is why oxygen and nitrogen has different categories based upon where it is. Definitely sulfur is one guy which is in cysteine and methionine, for that there is another. category. So, based upon that one, and there are some results which indicates this is the simplistic model. Using this one also we will get some good results. So, this is one software JRank. So, they use those kind of searching technique etc., what we have discussed, and this is their score function. This is a linear equation where this W indicates one weight value that is determined using neural network method, okay. And these E are the energy, where here you can see this is the van der Waals interaction, okay. This is the electrostatic Coulomb interaction, this is their dissolution definition, and they are using this one, okay. So, patch dock and fire dock, two other technique. Patch dock is okay, but for fire dock they have incorporated few more thing. One, you remember that I said that bound docking means this was there, you take it out trying to dock it, and unbound is this was there, you try to dock it. So, when it is the situation they are separately crystallized, I said the last stage possibly will be after identifying this you relax this one. So, that relaxation mechanism is there, side chain optimization does that kind of relaxation and tries to fit it properly. Okay. Well, so this is one technique when we do some algorithm development and implementation. So, before the testing we curate our data set in such a way that there is no bias, and we try to cover all possible corner cases. Okay. So, I am not going to details, if you are interested we can discuss offline. So, some of the success stories are like this. I am not sure whether you know or not. So, PDV, the protein data bank, is housing all the protein structures at the atomic level resolution. They are determined either by crystallography or by NMR, including cryo, EM or say solid state NMR. Now, for NMR you can get a functional structure, but for crystallography it is not guaranteed. For crystallography you may get some crystal artifacts. Okay. So, sorry, I do not have the structure of a crystal. So, for crystal what will happen? So, this is say one chain, this is another chain, then they will keep on repeating in a period, right. So, if it is the case, then this is one chain, this is another chain, then this chain will repeat here. Now, you can see that this guy has two interface or contact, one is this, another is this. Now, sometimes in crystal structure it is misinterpreted, because as such if you do not do any other cross linking kind of experiment, it is difficult to say that whether this is a valid or this is valid. Okay. Nowadays some softwares are there using which you can determine. Now, after this protein docking, so you can see that this is one previous structure, okay. This is one protein, this is another protein, this color indicates the compatibility, the color code is here, okay. And if it is compatible then only the matching is proper, okay. So, this indicates that matching is not proper, although it belongs in crystal lattice and reported in protein data bank, whereas the docking predicts that this is probably the structure, not this one. And here you can see that docking is doing good compared to the crystal structure. So, there are some success stories as well as some failure also, okay. Now, these are the other strategies where you can do some hierarchical scoring, or what you can do that if you have binding site information, say you are, so far I discussed the protein, but if you are working with say protein small molecule. So, you have one protein and you have one phosphate, you wish to do the phosphate binding, then most of the phosphate binding site is known to you. Then instead of going for all possible decoy generation, you can just focus only on the phosphate binding site and try to bind there, then definitely it will be very fast and you can do some sophisticated calculation also. Well, so in this age of high performance computing or supercomputing, so you can go to parallel implementation for that one. How we can do that one? Look, it is mutually independent. So, given two protein, so you can ask one say computer or processor or node that you generate all the translation cases. You can say another, you generate all the rotation cases, or you can say that you do only x axis translation, you do only y axis translation. That way you can divide the workload among all the available processors, and that way it can be very fast because it is embarrassingly parallel. So, there is as such no dependency between two different decoys. Only thing is that you have to generate the decoy, you have to evaluate the decoy separately, and then you combine the result. Okay, so parallel implementation is possible. Well, so it was the docking, and from that point of view it is interaction. Now, people is moving little further. So, what they are trying to do, that if I have the knowledge of more than one interactions, then whether I can make some holistic picture or make some idea out of all those interactions or not. And if it is, then probably one of the best representation is some graph based representation. Okay. This very simple thing, graph is defined by these two things, one is called V, another E. V is the set of vertices, E is the set of edges. Okay. Now, these are the vertex, and edge means when there is some connection. Now, this connection may not be a straight line, it may be some curved line also. Okay. And using this curved line, what you can do? You can get one graph. Now, look.
So what does that mean by curved line and straight line? I mean, what is the meaning of that?
No, this is just to clear out the confusion, that if it is curved line, then also it is edge, if it is straight line, then also it is edge. Okay. Now, for this particular graph, what you can see, that what I am telling, this is vertex, this is vertex, this is vertex, this is vertex, these are vertex, and these are the edges. Edge can be straight line or curved line. Okay. There is no differentiation between these two. Now, for this particular graph, what you can see, that from any vertex to another vertex you cannot reach, right. This is kind of one island. So, this graph is called as the disconnected graph. Okay. But if I add more edges, then it will be a connected graph. Okay. Now, regarding the properties, in this graph the edges are there, but there is no such direction, indicating from here I can go this way, from here I can go this way, both are feasible or possible. So, this is called as the undirected graph. Next is degree of a vertex. So, vertex is all those circles. The number of edges which is connected with that vertex is called as the degree of the vertex. Okay. You can count that and you can have that one. Another point is that shortest path. So, from here to here, what is the shortest path? I can reach 1, 2, 3, I can reach 1, 2, 3, 4, 5, I can reach 1, 2, 3, 4, 5, 6, all those possibilities. What is the shortest path? Yes, that is one. So, the advantage of doing all those things are that, given a set of interactions, if you make a graph, then graph theory is a separate discipline where people are working only on the graphs. You can probably have one algorithm to infer something out of the graph, right. I will come to that one. Now, center of a graph, that is very interesting. The eccentricity of the vertex in a graph is the greatest distance from V to any other vertex, right, and the vertex with minimal eccentricity is called as the center of a graph. Center of a circle is what? The point which is equidistant from the all the points, right. So, in this case also, which is minimal, that is going to be here. So, in this diagram all the red circles are basically the vertex. Interestingly, there may be one or maybe two vertices for a graph which acts as a center. There are proofs, we are not going into details of that one. Well, so last time I did not write A, B, C, D, etc. Now, I am writing, and when I am writing I can call that as a labeled graph, and frequently we may need to label the graphs. Okay. So far there was no weight assigned here, now I can add some weight, right. When I add weight, then that is called as the weighted graph. So, weight indicates that the amount of, if there is some cost involved for going from here to here or for something, then I can add that one, and then that way I can say it is weighted. Now, once a graph is weighted, then I have to redefine the shortest path. Probably that shortest path is not shortest path here. You look at that shortest. So, initially you said this was the shortest path, right? Now, the amount of weight is 4 plus 5 plus 3, 12. Now, let us explore something else. So, 4, 5, 6, 8 and 3, 11. So, this is the shortest path now. Okay. As of now, there is no direction. I can add direction also, indicating from B I can go to H only. I cannot go to A or I cannot go to F. I have to follow the direction only, right. Well, when there is a directed graph, I need to redefine degree. Now, degree is either in degree or out degree, means the number of edges which is going out of this vertex or the number of edges through which I can reach to that particular vertex. Okay. In biology, we can use this network for several reasons: protein-protein interaction network, metabolic network, genetic interaction network, transcriptional regulatory network, cell signaling network, blah blah blah. Okay. So, let me explore only protein interaction, and I think you can map it for other cases also. The definition will be like this: given a set of proteins and their pairwise interaction information PPI, construct a graph where each protein is a vertex, and if there is any interaction between two proteins, then there is an edge between two corresponding vertices. Very simple. Okay. So, once you will have the graph, then you can use some topology analysis, like you can do clustering, you can check that which node is connected with how many degrees. Okay. So, using that you can go for the priority of that one. So, here one example, let me elaborate this one. So, this is one protein interaction network. It says that this is EVP virus and human interaction network, right. Now, here as per the definition of our protein interaction, these are proteins and there is an edge between these two proteins because these two interacts with each other, right. Now, look what is the advantage of having this one. If say I wish to design one inhibitor or if I wish to design one drug, so that we can heal this this EBV virus, then we can target some protein, right. Now, when we are targeting the protein, if I do not look the, or if we do not have the holistic idea of its interaction, then I can design one drug which will basically block the function of these three proteins, and that way we will have some side effect. On the other hand, if we know that that particular say BPLF1 protein interacts with so many other proteins, then you should look each of the proteins binding site and then design one inhibitor or drug in such a way that all the essential functional remains, rest of the functions are killed.
That is virus protein.
Which one?
That one is virus protein.
Yes, right. Right. Also, it has another application, although the field is not developed, that if I wish to increase the interaction, then I know the binding site, then if I wish to increase the interaction, then which kind of mutations I will allow and at which position. Okay. At the if I zoom in, then what I can see that basically they are different proteins, because these vertices are nothing but the proteins. Okay. So, here is another application. What they are doing, they are combining gene expression data, PPI interaction network data, signaling pathway. After doing data analysis and integration, candidate disease genes, disease network and disease pathways are identified, and this helps in biomarker discovery. But after that one definitely you have to do some kind of experiment. Okay. And why we need to go for protein docking algorithm or say parallel implementation of that one? Because as of now these 4 are the experimental PPI databases, okay, which contains the protein interaction data, not all at the atomic level resolution, but to some extent if they have the data from there we can integrate those data in order to generate the protein interaction network. But look, in the protein data bank as of today 140K protein structures are available. Theoretically in situ possibilities, so this many number of interactions are possible, all are not tested, okay, and it is not possible at least in our lifetime to test all of them. Probably we have to go for some computational technique for that one. Okay, that is it. If you have any question you can ask me. Okay. So, these are my lab members, 1 postdoc, 5 PhD, 1 MS. Okay. Thank you.
You explain the weakness of the graph not